Hello, welcome back. Good morning, or good afternoon, or good evening, wherever you might be in this world. Um, Today is day two of our online Chime 2 workshop at the FAES, and we're really excited to keep on going here. I just have a couple of announcements that I wanted to go over before we jump into the first lecture of the day. Um, and also, really quick, I'm just going to ask the someone on the instruction crew if we haven't pinged the whole um, announcement section or everyone on Zulip. Can someone please do that right now? Um, so yeah, yesterday we covered a lot of sort of initial material related to Chime 2, so some of the project history and information about sort of some of the language and concepts of Chime 2. We also got a chance to log into our workshop server for the first time and run our first Chime 2 command. And we talked a lot about sample metadata and about the tutorial data set that we're going to be working with this week. So yesterday really was like a lot of foundation work. And then starting today, we're really going to get into kind of the nitty gritty details. So we're going to be covering some 16S lab protocols, importing data, and denoising methods, and phylogenetic reconstruction. And so we're going to have individual lectures on all of these topics, and then we will also have some hands-on tutorial work as well. The other thing that I just wanted to mention really quick. So I shared an announcement earlier this morning with a document that attempts to show how to configure your notifications and send and receive private messages. If the control room can make sure that my screen share is on, I just wanted to kind of go over some of that here in person in case anyone wanted to follow along with me to customize your notifications. We good, control room? Cool, thank you. So I have loaded up here a demo account that I've created. So I'm logged into my demo account, and it is signed in at workshopchat.chime2.org, and I'm just looking at this here in my browser. And if I wanted to customize my notification settings, I can move over to this upper right corner. There's a menu with a gear icon. And if I click on that, I can then press the settings option. And this will open up my demo account's settings. And I can click on notifications over here. And there's a nice grid with all kinds of notification settings that you can configure. So you can choose what kind of notifications you get for private messages. That's what PMs stand for. As well, mentions and other alerts. So mentions are when you @ someone. So I could say @ Evan Bully, and how do you accomplish this this concept or this task. So you can customize if you get audio or visual notifications for all of those. And also emails. And then the streams is a separate set of settings here. And streams are the things over on the left-hand side of the screen. So I'm going to close this menu. This is configured as I would like. And I just want to point out this over here on this left-hand side, there's a couple of pieces of information. There's messages and private messages and mentions and so on. And then there's also the streams listing down below. So all messages is showing that I have 16 unread messages up in here, and they're just kind of interleaved all of the different private messages and all of the different public messages that this user account has received. It's showing which stream those are in and then which individual topic within those streams. So you can see there's some announcements and so on. And as I scroll through and read these, you can see my unread count in this is updating. Similarly, if I wanted to look at my private messages, I can click on private messages, and see all of the private messages that I've received. So one of them is from this Zulip welcome bot. And then I also had a discussion with my my instructor account as well. And so if I wanted to filter down to just that private message between the instructor account, I can click on that over on the left-hand side, and it shows just a private chat here. And the thing that I just want to point out here is this is how instructors will be reaching out to you if you get stuck during the tutorial. So if you post in discussion and say, hey, I'm stuck, I'm getting an error message, and an instructor says, okay, let's hop on a video chat, they're going to send you a message, a private message with a link to a video chat. So it'll be really important for you to keep an eye on your private messages over here and make sure that you let the instructors know when you've received the message, and if you've solved the problem, let them know. Otherwise, coordinate with them to hop on a video call. They will be able to set that up for you by creating a link for you. Then I think that's it. That's all I wanted to cover with the notifications. I'll just check in with the control room real quick. Do you have anything you want to add? All right, so with that, I am going to switch on over here to the first lecture of the day. And so this is by Emily Borsum. Emily is a PhD student in the COPE lab at Northern Arizona University. And Emily has put together a really nice primer on 16S lab protocols. So sort of all of the stuff before you get to QIIME 2. So getting samples ready for sequencing and so on. So with that, we are going to cue that video up. And also, I'll just make a quick note that volume can be a little tricky on this one, so you might need to turn it up a little bit. There's some background noise in this video. So thank you.
The first step in our 16S rRNA gene sequencing flow is to extract the DNA. Today we will be looking at two different types of DNA extractions. The first is called the Kingfisher extraction, where we set up plates and place it on a machine, and the machine uses magnetic beads to extract the DNA for us. The second type of extraction we will look at today is the QIIME all-prep extraction. This will extract both DNA and RNA and elute them separately. For our extractions, we use a biosafety cabinet to keep all contaminants out. Even though our samples are considered BSL-1, which means they are not harmful to humans, we keep them inside the biosafety cabinet to protect the samples themselves from bacterial contamination. To do this, we use our PPE, which is our protective equipment. So here I have a temp lab coat, as well as gloves, tape, and I will place one more pair of gloves over. Our first step is going to be to make the lysine binding solution. This will be placed in the lysine matrix E tube so that we can lyse the cells and release the DNA for extraction. So first we will take the lysine binding solution concentrate. And add this to a 50 ml conical. Normally we would add more than this, but for video's sake and for ease and clarity, I'm just adding a little bit. We then add the carrier RNA. We will then vortex this to mix. Our next step is to prepare the bead mix. These beads are the magnetic beads that will help the Kingfisher extract the DNA. We'll begin with an empty 1.5 ml sterile tube. I will we will add a 50-50 ratio of nucleic acid binding beads as well as a lysis enhancer. Before adding the binding beads, it's suggested that you invert to mix up the beads. Now that we have our mix, it's suggested to vortex before putting away in the 4 degree if you're not going to use right away. While one person is making the bead mix and the lysine binding solution, the other person can be working on the plates. In total, we will have 6 plates for this extraction. 5 of them will be deep well, and the final one will be a 200 microliter plate. The first 2 of the deep well will be wash 1 solution. The second 2 will be wash 2. And the final plate will be the sample plate. The last plate, which is the 200 microliter plate, will be the elution buffer plate. So to prep all of these plates, we take 1 deep well at a time, add our wash concentrate. Before starting the extraction, you should add the isopropanol as suggested. And then multi-channel pipette from the boat into every single well in this 96 well plate. Now it comes to the sample. So the sample will be added to the lysine matrix E tube along with the lysine binding solution. This will then be homogenized and spun down to leave a supernatant, which is a layer of liquid above the beads. This supernatant, also known as the lysate, will be transferred into our sample plate. This is the sample itself. Our next step is to add isopropanol. And finally, the bead mix. Normally we would do these 1 step at a time for all 96 wells, but again, for simplicity and clarity, all 3 are added to 1 well. Our final step will be adding all of the plates to the Kingfisher machine for the extraction. From here, we're going to load the Kingfisher machine. So once you have your protocol loaded onto the machine, it will tell you the order of the plates and when they go on. So our first will be the tip plate. These tips are what attach to the magnetic beads and carry the DNA and RNA through the extraction. Always make sure to line up A1 in the correct corner. Next will be our elution plate. And wash 2, and so on. Move on to our wash 1. Again, making sure that A1 lines up in the correct corner. And finally, our sample plate. From here, we press start. The run will take about 30 minutes, and once it's complete, you can come back and take your samples that will have both DNA and RNA eluted into the same one. This next extraction is the QIIME all-prep DNA RNA extraction. In this extraction, DNA and RNA are eluted separately. So we have two columns. The first is a pink column, which is an RNA column tube, and the second is a white and purple, which is our DNA column. In a column extraction, the nucleic acid, whether it be RNA or DNA, is caught in the column, and we wash any impurities such as the opposite nucleic acid or proteins through the column so that our final elution is pure RNA and DNA. We add both our tissue and the buffer RLT plus into a lysine matrix E2. My tissue is already in here, so I'm going to add the buffer. Once the sample is centrifuged, we will add the supernatant into the DNA spin column. Once we've added the buffer to the spin column, we're going to centrifuge it down to push the fluids through the column. We will do this after every step. Our next step is to take the DNA spin column, which by now has trapped the DNA, and place them into new collection tubes. Save the flow through for RNA purification. From here we will add ethanol, mix the ethanol and flow through. And transfer the mixture into the RNA spin column. First, we'll go through the genomic DNA purification step. With our DNA column, we will add our first wash buffer. We will then spin down this column to produce the flow through. From there, we would release the flow through into a trash can and continue with our AW2 or our wash tube. We would repeat the same steps, spin it through, discard the flow through, and our final step would be the elution step. In the elution step, we add our buffer EB. We would then spin this down and transfer any flow through to a collection tube as our final DNA product. After the DNA extraction, we need to verify the quantity nanodrop, which is a spectrophotometer. This uses the absorbance to calculate both the quantity and quality of the DNA. First, we will load the DNA samples onto the nanodrop spectrophotometer. We do so by pipetting 2 microliters of the DNA directly onto each pedestal. From here, we will choose DNA and click measure. From here, it will give us the concentration as well as the 260-80 and 260-30 ratio. Based on these numbers, we will be able to determine the quality of the DNA. Once we have ran all of our samples, we can take a look at the data. We would expect our concentrations to vary depending on sample type. In this case, we're looking at fecal samples, and we would expect our concentration to be high. We always want our control to be the lowest concentration, otherwise we might be concerned that there's contamination. After we have verified the quality and quantity of our DNA using nanodrop, we will run polymerase chain reaction, or PCR. We use PCR to target a specific gene and amplify or make many copies of that gene. In this case, we will be amplifying the 16S rRNA gene. To begin, we will decon all of our materials needed and place them into a PCR hood. We use a PCR hood to keep everything that we want in the hood and everything that we would not want, such as contamination, out. To begin PCR, we will decon a PCR hood and decon everything that we place into the hood. From here, we can turn on a UV light to remove any contamination, such as bacteria, that may be present still on our materials. Our first step will be to make a master mix with all of the necessary reagents for a PCR reaction. We'll begin with DMTP mix, BSA, 10x PCR buffer, a forward primer, and tap. We will then add all of these into our master mix tube. Next, we will begin to load our PCR plate. In each well will be a different reaction containing the master mix, the reverse primer, and the sample. We will run each sample in triplicate and include a no template control. Now I have my master mix, DNA, and reverse primer combined into each reaction well. I will finish by sealing off the plate with a PCR seal, spinning it down in the centrifuge, and adding it to the thermocycler. Our last step of PCR is to place the PCR plate on the thermocycler. The thermocycler will use heating and cooling cycles to amplify the 16S rRNA gene. Our next step in the 16S workflow is going to be to run a gel electrophoresis. Here, we're going to take our PCR amplicons that we just finished the 16S PCR with and dilute them with water into another plate, and from here we will add them onto the gel. The gel will help to separate them out by charge as the DNA travels, and we'll use a ladder to track at what length the base pairs are. First, I'll begin by adding the water to the dilution plate. We always start with water because it is a lower risk of losing over the PCR amplicons. Next, I'll add the amplicons. Next, we'll load our gel. Because the gel has ethidium bromide in it, we need to wear goggles. From here, we just gently tear open. And being careful not to rip the gel wells. And here you can see the different gels and each of the wells. And what we'll do here is we'll load each well with a different sample. We will load both our amplicons and our NTCs, which if you remember is our non-template controls. And from there we will run the gels, and as the DNA moves, it will show how far and the length of the band. First we'll load our PCR amplicons. And then we'll add our ladder. Next, we'll load the gel. While this isn't the gel that we ran, this is a really good gel to take a look at. So here we can see the ladder is run on both the right and the left side of the samples. And on the odd wells, we have the PCR amplicons, and the even wells have the no template control, or NTCs. So what we expect to see here is that the odd numbers have nice bright bands, and our NTCs shouldn't show any bands at all. We expect this bands to occur around the 380 base pairs. And as we can see here, it's just between the 300 and 400 base pairs. So exactly where we would expect. So these here are 16S rRNA gene PCR amplicons. To recap what we did today, we took our samples and extracted DNA. Following that, we quantified the DNA using nanodrop and used PCR to amplify the 16S rRNA gene. We verified these amplicons using an e-gel. We quantified the amplicons using QIIME. And finally, pooled the amplicons, cleaned them up, and sent them off for sequencing. Thanks for joining this section of the QIIME 2 workshop.
All right, thanks for that, Emily. So I heard from the COPE lab earlier this morning. They are going to be joining us this afternoon for Q&A. So feel free to send questions for them over into discussion, and we'll go ahead and hold on to those for this afternoon's general Q&A session. So we will not hold a specific Q&A about this topic right at this moment. We're going to move into our next lecture here, and this is from Mehrabad Astaki. He is a postdoctoral researcher in the Night Lab at UCSD. And Bod is going to walk us through importing sequencing data into QIIME 2 and sort of some of the things to think about related to multiplexed versus demultiplexed sequencing data and so on. And then Bod will be joining us after that for some Q&A. So thank you.
Hello everybody, my name is Mehrabad Astaki. I'm a postdoctoral researcher in the Night Lab at the University of California, San Diego. In this video, I'm going to give an overview of the process of importing data into QIIME2. We're going to be mainly focusing on importing raw sequence data and also demultiplexing of these raw reads. In this video, I assume that you're already familiar with the core concepts of QIIME2 that was covered in a previous video, especially with regards to the semantic types and the file formats of QIIME2 artifact, as these become pretty important component of importing files into QIIME2. Okay, this is a basic overview diagram of a simple workflow in QIIME2. This workflow starts at the top left with raw sequences and goes through the demultiplexing of sequences, denoising of our reads to form a feature table and representative sequence files. Then it creates a phylogenetic tree as well as a taxonomy file. And finally, it runs some diversity analysis and to produce some nice results visualizations. What is nice about QIIME2 is that you can import your data at virtually any of these steps in the pipeline and continue to use the various available plugins downstream without the need for the upstream files. So, for example, if a collaborator gives you a biome table that they have produced elsewhere, say in R using a new in-house pipeline, then you can simply import that biome table without the need to have to access the original raw sequence files. And you can just work your way downstream through the available plugins and analyze the data using the new biome table you've imported. However, before you can do that, you need to import your data into QIIME2 in the correct format. That is to say, you need to know the type of file that you have as well as the format, if needed, in which it was made in. In this series of videos, we're covering the entire process starting from raw sequences. So in this video, I'm going to mainly focus on describing the importing process from raw sequence levels, but I just wanted to re-emphasize that importing can happen at any of these steps. So before we start, I just wanted to give a disclaimer here, and this is purely my opinion based on my experience working with a variety of data types, previous QIIME2 workshops, and answering questions on the QIIME2 forum for the last few years. And I found out that the importing of raw step is often the most confusing part of the QIIME2 pipeline for new users. I've personally found this to be true with any bioinformatics software that I've used, so this is definitely not exclusive just to QIIME2 by any means. And the main reason why this step can be confusing is that there are many ways your data can exist in tens to hundreds of variations that exist to date, and only one of those is the correct one for any given data. So you really need to know what type of data you have and in what format it's in, and so this is often a difficult concept for new users who may not be familiar with this type of data. And just so you don't think I'm exaggerating when I say there can be hundreds of different data formats, this is a list of 46 importable types and 75 importable formats, which my QIIME2 environment currently recognizes. And of course, there can be even more depending on if you have any additional third-party plugins, QIIME2 plugins installed. And I show this not to intimidate you, but rather to reassure you that if you do find yourself struggling with this portion of the pipeline when you're analyzing your own data for the first time, just know that you're not alone. And the good news is that once you have imported data into QIIME2, basically the hardest part is over, and everything downstream is much simpler and easier to find help with. Now, while there's no automatic way of detecting your data type and format, there is at least one resource that I know of that may be useful in helping you make sense of your data. This is an example of an excellent quick reference flowchart made by Nick Bakulich. This can be found on the QIIME2 forum in the link I provided here, and this can help identify which type and data format of your input files may be in majority of cases. Perhaps not all of them, but it will do a pretty good job of for most of the cases. Okay, now let's take a closer look here at the process involved in importing your raw sequences into QIIME 2. So far up to this point, you have completed your carefully designed experiment. You have collected your samples and extracted DNA from them. You've amplified your target gene of interest, for example, in this case, the V4 region of the 16S rRNA. You've added unique barcodes to the reads from each sample, and of course, ever so carefully recorded those per sample barcodes in your metadata file. Next, you'll pull all of these different samples together and run it through your sequencing machine. The sequencing machine then performs its magic and gives you some output in the form of FASTQ files. I'll describe FASTQ files in more detail a bit later, but essentially these are the files which contain the actual sequence information of your reads. You'll have one FASTQ file that holds information about the actual sequences, and another FASTQ file that is specific to the barcode sequences. For simplicity's sake, this example is just demonstrating sequences of the forward reads only, but if you have paired end data, that is, if you've sequenced the reverse reads as well, then you will receive an additional FASTQ file that holds information on the reverse read. So what is a FASTQ file? Well, you can think of FASTQ files as essentially a text file that holds various information about your sequences in a somewhat standardized format. These files are built on the older FASTA format that have been around for many years, most popularly perhaps used with the 454 pyrosequencing platform. And the major difference between them is that FASTQ files hold additional information about the quality of each base call. This is in fact where we get the Q in FASTQIIME, which tells us that if this is a FASTA file with quality scores. In a FASTQ file, each read is described in exactly 4 lines. The first line, that starts with the @ symbol, is a sequence identifier. This line is not really something that is well standardized. So what is written here can vary from different sequencing facilities, but generally holds some information about the run, the equipment ID, the run ID, lane number, perhaps the date, and so on. The second line is your actual sequences. In this example, this is our DNA sequences from our amplicons, but of course if this was our barcode FASTQ file, for example, then this would simply correspond to the 7 or 8 nucleotide long unique barcodes. The third line, denoted by the plus signs here, is a placeholder line, which can technically hold a variety of information that you may want to include, but these days you mainly see just a plus sign indicating that this is a placeholder line. And finally, the fourth line holds quality scores corresponding to the sequences. These quality scores are coded using a series of ASCII characters, which are then translated into numerical values downstream. These quality scores, also known as Phred scores or Q scores, are calculated by the sequencing machine, and they tell us about the quality of our nucleotides in terms of error probabilities. So, for example, the question mark character corresponding to the ASCII code 63 here translates to a quality score of 30. A quality score of 30 indicates that the probability that the corresponding nucleotide being incorrect is 1 in 1,000 or is 99.9% accurate. In other words, if we saw a G, for example, nucleotide in our reads, the likelihood of that G having been called G by error, and in fact it was meant to be, let's say, a C, is 1 in 1,000. And these quality scores are very important and become a crucial component of our quality control steps and filtering steps that you'll learn more about in later videos. Okay, now that we have a better understanding of our FASTQ files, let's go back to our example data. So again, here we have on the left our sample metadata file, which contains information about each of our samples, including their unique barcodes. In the middle, we have a FASTQ file for our barcodes. And on the right hand, we have another FASTQ file for our actual sequences. What is important to emphasize here is that at this point, our data is still multiplexed, meaning that all of our sequencing data is contained in one location, in one file, and they are not linked to their original sample source yet. And what we ultimately want is to group all of our sequences from the multiplexed FASTQ file, and we want to demultiplex them so that each sequence is paired with the sample it originally came from. In other words, we want all of the orange sequences to be paired with our orange sample, the blue sequences with the blue sample, and so on. And the way we can achieve this is by simply mapping the sequences back to their sample of origin using those unique barcodes that we added at the beginning. So this is a very simple overview of how the demultiplexing process actually works. We start from the right with our sequence FASTQ files. We take the first read we see there, and now we move to the barcodes FASTQ file and match it with the first read we see in that file. It's worth pointing out that the order of sequences between these two FASTQ files is paired and are always matched when they are produced by the sequencing machine. So what I mean is that read number 1 in our sequence file will always correspond to read number 1 in the barcode file. Same thing with read number 2 in the sequence file will always correspond to read number 2 in the barcode file, and so on. Now in our barcode file, we read the unique barcode identifier that is associated with that read, and finally we can map that barcode using our metadata file and identify exactly which sample that barcode corresponds to, so meaning what sample our original read came from. In this case, the orange sample was our source, our original sample source. So we repeat this demultiplexing process for each read until all of our reads have been assigned to one of our samples. When the demultiplexing process is complete, instead of having one FASTQ for all of our samples, we'll now have one FASTQ file per sample. And we no longer need our barcode file because we have already extracted information that we used from them. So this is what we refer to as a demultiplexed files. And of course, when you are working within the QIIME2 environment, you will only actually see a single QIIME2 artifact. However, we now know that the underlying structure of that artifact is a series of individual FASTQ files. In fact, if you were to export this artifact at this point, you'll see that all of these FASTQ files exist as a separate file within it. And of course, again, if you have sequenced paired end data, then you will have two FASTQ files per sample, one corresponding to the forward reads and the other to your reverse reads. It is also worth mentioning here that at this point, that if you have paired end reads, your forward and reverse reads are still not joined together. That is something that happens at a later step. At this point, they are simply held, paired together, but as separate files. Now, depending on the sequencing facility where your data came from, you may receive your data in either multiplexed form, such as in our example here, or demultiplexed. So if you receive multiplex file, then of course you will need to demultiplex your reads. But if you receive them already demultiplexed, then you can simply skip this demultiplexing process in QIIME 2, and you can just move forward to the next step, which will be denoising and clustering. The easiest way to know if you have received multiplex or demultiplex data from your facility is by simply looking to see if you have received one FASTQ file that contains all of your samples, or if you have separate FASTQ files for each sample. So this concludes the lecture tutorial on importing and demultiplexing reads into QIIME 2. In the next section, you will get to actually get a hands-on experience on how to import your raw FASTQ files in QIIME 2. And we'll see you again at the next video in this tutorial series, which will be about denoising or clustering your data. Thank you very much for joining.
All right, thanks for that, Bod. I am joined here in the control room by Mayor Bod Astaki, and I think Bod has a couple of minutes to take some questions. I have one that came up while the video was playing about the one of the examples in the slide deck. So Bod, this is over in the questions broadcast. So someone wanted to know, why can't I see the barcode sequences in the first or last string or the matched sequence in the slides? And I am just pulling up the slide deck over here to get my hands on the right slides. Let me see. Actually, could the person who asked that question, can you follow up and let us know specifically which slide you're looking at? That'd be really helpful. And if anyone has any other questions, feel free to send them into the discussion channel right now. And if you don't have any at the moment, but you think of them later, we can always queue them up for the general question and answer session at the end of the day. Bod, I guess while we're hanging out here for just a minute, I'm going to ask you a question that I asked Greg yesterday. How did you get started in scientific computing or microbiome, or this kind of topic of this workshop here?
Good morning to everyone, by the way, if it is your morning. And sorry about the slide follow-up question. I actually had the same question. So if the user who asked that original question can lead us to the exact slide number, I'd be happy to help him with that. So I got into bioinformatics, microbiome data analysis specifically, sort of out of a necessity. My background in the sciences came from kinesiology of all streams, and I really did my first round of microbiome analysis when I needed it for a paper that I was working on that sort of connected exercise and microbiome. And it sort of just started from there, where I became interested in how analysis were done, because I didn't like the sort of pre-packaged ready summary of the data that our core facility would give us. I thought it just wasn't deep enough. So that's kind of how I got interested in the first place. Just decided to go for it and try to dig deep into data. And I'm very lucky. I'm very happy that I did, because that, proved to open up multitude of doors for me in my career. And so once I caught the bug of just looking through data, I had never written a line of code in my life. I never had done any programming, any sort of computer programming whatsoever. And so I started reading on the QIIME 2 forum, just
reading every day as sort of a procrastination, really. And I kind of got interested just with a variety of data people had, a lot of different questions people had, and I started learning from it. And eventually, I think I had seen questions pop up often enough that I thought to myself, hey, I could help with that. I've run into the same problem millions of times. And I think eventually, after a while, the QIIME2 team saw my name so often on the forum, and they said, okay, we gotta talk to this guy, see what he's about. So I really got lucky there, and they invited me to join a QIIME2 developers workshop. That was super fun, and that's how I got to meet the whole QIIME2 team, a lot of developers, and again, that sort of just fed into my whole interest in the microbiome world. And so I kind of got involved just by happenance in that way, and I can't stop now. I really enjoy the microbiome world. Since then I've gone into a lot more programming. I do a lot of my work in R, trying to learn a little bit more Python. And so just from that one simple step of procrastinating every day a little bit on the forum, it turned into a whole career almost here.
That's really awesome. Thanks for sharing. And we're really glad to have you here. Looks like that person followed up and let us know they're specifically asking about slide 122. And let's see. So the question is, why can't I see the barcode sequence in the first or last string or the matched sequence? So I'm looking at the slide deck here, and the first thing that I just want to point out is there's the color coding to show which barcode. Let me see. Can I? Sorry, I'm getting messages from the control room here. Let me share this here. Can you see this? So this is slide 122. And so what I'm looking at here is we have our table of our sample metadata, and you'll notice we've color coded each of the samples listed in here. So there's 4 samples, each has its own unique barcode. There's the yellow, blue, reddish, and purple. And in this middle column here, we have the barcode sequences themselves. So this is an example representing an EMP style formatted multiplex data. So this is where you would have the barcode sequence in one file and the actual sequence in a separate file. And so the way that you read these is, so you come through and you look at this first row and start with the barcode file and look at the barcode sequence. And so you'll see here AACGCAC matches our sample 4AC2. And so that's why this is colored yellow here. And then that corresponds to the first row of sequence data over here on the right-hand side in the sequences.fastq.gz file. And so that means that this particular record belongs to the first sample. And then similarly here we have the second sample back to the first sample, the third sample, and the fourth sample. So if you look through here, each of these barcode records, they do wind up matching back up to a barcode sequence in the sample metadata. Pull up my chat really quick. So I'm going to turn off my screen share here. So Bob, there's another question that came in about reverse complements. Let me find that here. So when do we need to care about taking a reverse complement of barcode sequence, and what kind of considerations do we need to have in mind for that?
Yeah, that's a good question. I wish there was an easy way for me to say, this is when you need to do this, is when not. But the reality is that every sequencing facility and kind of does things a little bit differently. So the answer kind of, again, really depends on what kind of data you've been given, how the experiment the sequencing has been set up. One way you can check though is by simply looking at the raw FASTQ file, whether it's your barcode files or your actual DNA FASTQ file, and you can because they're basically a text file that you can read. You can just take a look at them and see what the actual sequences read, and check that manually with your mapping file or your metadata file, whichever you have, that describes the data that you've come up with. So if you do see that the sequences are not matching, likelihood is that they've either been reported as reverse complement, in which case then you would need to tell QIIME to do that transformation. And there's a series of parameters in QIIME2 which we might, I can't remember if we do cover in the hands-on portion, but there are parameters that you signal to QIIME2 that says, okay, that my data is either my sequences are need to be transformed, or you can tell it to convert your mapping file to do that transformation. So definitely important, because otherwise it's not smart enough to know the orientation. So you definitely do need to give it that directive. But is there an easy way to tell or be sure? No, you need to either know your data or look through the data.
Yeah, and I'll echo, the asking the sequencing center is always a really good idea too. If you have any questions about, is my data multiplexed or demultiplexed, or, how are my barcodes situated in this data? It's always really helpful to ask the sequencing center. So let's see. I'm just taking a look at my chat really quick. So there was a follow-up question about that slide with the multiplex data. So the barcode matching in the QIIME2 EMP protocol demultiplexing steps only matches barcodes in the barcode sequence data. So you'll have two separate files, a barcodes file, and then either forward reads or forward and reverse reads. The matching process doesn't actually look at any of the data in the sequences themselves. It only performs the match for the barcodes.fastq.gz. So that might help clarify that a little bit there.
Yeah, if I can add, one thing that really helped me, I had a lot of similar questions when I was starting out, is that how does it know, the mapping between your actual FASTQ files and the bar. What's really important to remember is that the order in which those sequences are displayed are the same between your barcode file and your FASTQ, your actual sample files. So, in the slides that I had up, there were 3 columns. There was your metadata file, your barcode files, and your actual DNA sequences. And between the two, FASTQ files that you have, the order in which those sequences are spat out by the machine is really important. So if you, for some reason, delete one, even a single line in there, it'll mess up the ordering of all of them. So that's what connects those 2 files together, is the ordering in which they come. And then the recategorizing or demultiplexing is really done based on the barcode identifier.
Totally. Thank you. Got a question here from Dawn. If a sequencing error results in a sequence that does not match any of the barcodes that you used, at what point in the workflow is that noticed and dealt with?
Yeah, so there are, if you are demultiplexing your files, that's a step where you would decide whether you're going to keep a read or a sample or not. So if you have a sequence that has a barcode that isn't found anywhere in your mapping file, then that will be discarded. That's ignored because it didn't hit the required or one of the available barcodes. Now, there are some parameters that allow some form of error or some degree of error. So for example, if you have your barcode is 8 nucleotides long and only one of them doesn't match, so one nucleotide doesn't match, the next nucleotide. The next barcode, there is some leniency that you could direct QIIME to take. So you can tell it that, okay, allow 2, 3, 4% error, and that would usually means that you allow one nucleotide to be wrong and still be able to match to the closest marker. Now that has its own sort of limitation, and I would only ever encourage that if the barcodes have been designed with that error correction sort of mentality and having distances in place. But otherwise, you should sort of not allow it to have those mismatches. Otherwise, you're going to get some misclassified samples.
Great, thank you. We have another question here from Susan. What advantage, if any, have you seen in receiving multiplex data from a sequencing center versus demultiplex data?
That's a good question. So, of course, when you receive multiplex data, you have, as raw as the sequences can get in your hand, and you have a lot more control over what you're doing, and you know exactly sort of the parameters that have been used. So it's a lot easier to reproduce it. Whereas oftentimes, if the facility does that for you, you don't really know what they've done, what sort of parameters they've picked. So it might be hard for you to reproduce that in the future. Now, that being said, if you are in good relations and you understand the practice of your core facility, that might be a huge advantage, because the demultiplexing ultimately is just one of these long steps that you could go without if it's pretty standard in a pipeline. So yeah, if for example, as I was mentioning before, if your barcode sequences have been designed with this error correction option, where the difference between one nucleotide, whether by accident or not, doesn't reclassify your sample into a whole other sample. Sometimes, if your design is set up like that, it would be useful to demultiplex yourself, because most core facilities don't allow for that sort of leniency, that don't allow for that error correction. So in that situation, you may want to take control and do it yourself. But otherwise, it really is a matter of convenience to me. And I always make sure that I understand what my core facility is doing before accepting their demultiplex data as is.
Thank you. Yeah. We are going to move on to the next section here. So this is a follow-up to Bod's lecture. Chloe, a PhD student in the Capareso lab, is going to walk us through some of the next steps of our Parkinson's mouse disease tutorial. And so we're going to be importing demultiplexed sequences into QIIME 2. Before we cue that up, our control room is going to play a connecting to the workshop server video again. So before we get that going, make sure that you have a copy of your PDF cheat sheet right now that has your username and your password. And then we're going to open up Google Chrome and launch that Secure Shell app and get logged in. So we'll check back in briefly after that video plays, just to give everyone a minute to log in. So thank you.
First, we're going to navigate to Chrome: /apps. Chrome: /apps. Here you're going to see any apps that you have installed in your browser, and you should see the Secure Shell app here. So click on that. Next, you need to fill in your username and hostname. Hostname is workshop-server.qiime2.org. Make sure new connection is selected here. Go down here to username. Type in your username and a hostname. You'll notice it's pre-filling in text up here. You don't have to do anything up here. After you type in the workshop server name, all you have to do is press enter. Please note that all this text has nothing to do with your computer. This is actually related to only the workshop server and this Chrome Secure Shell app. Here you'll see a prompt for the password. As you type it in, you will not see any action here. That's for security purposes. It doesn't want to show your password. So don't worry if you don't see anything happening there. Just type in your password and press enter.
Hi, my name is Chloe and welcome to the section of the tutorial where we learn how to import data into QIIME 2. In this section of the tutorial, we will be running two major commands. The first one being QIIME tools import and the second one being QIIME demux summarize. The data we have for the PD mouse tutorial is already demultiplexed, so we don't have to demultiplex this data, but we are still going to run QIIME demux summarize to view our data. Before we get into all of that, I want to talk about where we are in our directories just to make sure we're all on the same page. So I'm going to ls in my user directory to find the workshop directory. It's really important that you're in this directory, so make sure that all of your data is in the workshop directory. So I cd'd into the workshop directory, which means I changed directories. And then I'm going to ls in there, and I can see my mouse_tutorial directory, which you guys should have made in one of the previous sections of the tutorial. And I'm going to cd into that mouse tutorial. And now I'm in there. So let's check out the data we have in the mouse tutorial directory. You have both of your metadata files. You downloaded metadata and visualized it in the last section of the tutorial, so there's your data from that. Now we are looking to import our data using a manifest file. Importing data is probably one of the hardest steps, but we need to get our data into QIIME 2. So in this section of the tutorial, we'll talk about the type and the format needed to import demultiplex data. Importing demultiplex data into QIIME means that we don't have to demultiplex it, but it does mean that we have to use a manifest file. Some data does not come already demultiplexed, and we have to run QIIME demultiplex in order to demux in order to demultiplex it. If you want to see steps on importing demultiplex data and running QIIME demux, I would encourage you to check out the moving pictures tutorial. Okay, so let's get into running our commands. So first off, we are going to download some files from the internet. My first one, I'm going to use wget, and I'm going to copy and paste that all over here and enter and run it. And then I'm going to run the command ls to see if that data actually downloaded like it said it did. And we can see that it did, so hooray. Now let's go to the next section where we're downloading our sequences. I'm going to do the exact same thing I just did. Copy from that little guy right there, paste, and enter. Awesome. So our sequences are amplified using an Earth microbiome primer, and were sequenced on Illumina technology. They are 150 reads, base pairs reads, and so our sequences will be slightly too short to do paired end read analysis downstream. They're too short basically because they aren't long enough to have sufficient overlap, and that means that we can't use paired end reads. But we can use single end reads, so we're good to go. We should have just downloaded our data, so I'm going to check, and we can see that we have a demuxseq.zip file. So a zip file is compressed, so we can't access it like that, and we'll need to unzip it. Luckily, there's a command, unzipped. So I'm going to copy from this little guy over here. I'm going to paste it right in, and you can see all of the sequences that were in that zip file inflating. And now if we ls, we can see that we have a directory called demuxseq, which have all of our sequences in them, which is perfect. So now we are going to take a look at our manifest file. I'm going to go a little off script here. The tutorial says to use head, but I'm going to use nano so that we don't get that line wrap effect and it's just a little bit clearer. So let me zoom out just a tiny, oh, not what I meant to do. Sorry. I don't understand what happened. I'm going to exit this and try again. Okay. We're going to, okay. So I just am trying that again. I think I just kind of, my computer just kind of freaked out a little bit. But, so now we have our manifest file. So I exited, sorry, I just want to back up a little bit. I exited using Control X, and then I just copied the nano manifest file so I could see it. So now we can see our file. Our manifest file is a tab separated comma. There's one column called sample ID, which has a unique identifier for each sample right here. And then there's the absolute path, which tells us where the sample is located. And that's basically what a manifest file is. If you had paired end reads, you would have another absolute path called the reverse path, and this would be the forward one. But since we just have a single paired end read, we just have one absolute file path. So I'm going to Control X to exit out of that. I'm going to zoom in a little bit because I like to be able to see what I'm typing. And we are going to actually import the data. So we're going to scroll down to this QIIME tools import.
We've had a couple of folks let us know that they are just needing a few moments to catch up, so we'll do that. I'll just remind everyone the links to the workshop tutorials and so on are over in announcements under workshop slides and other handouts. So I will go ahead and forward a copy of that right now into the stream here. So there should be another copy of that. Oh, looks like Ryan Thomas beat me to it. Thank you. So we'll just give everyone a second to pull that up and then scroll down to the section right after the sample metadata that we finished yesterday. Let me share my screen really quick. Okay, so I am going to copy and paste that URL and load that up in my browser. And I'm going to scroll down a little bit here. So yesterday we covered sample metadata. And today's section is starting here at importing data into QIIME 2. And so our first step here was downloading our manifest file, downloading the demultiplexed sequences, and then unzipping. If you ran something like nano instead of head here to look at the manifest file, you can hit Control X to get out of nano, I believe. If you open it up in less, you can use Q to get out. So we were just looking at the manifest file here to understand what samples and the files associated we were working with. And then the next step is this importing command. So I think we will go ahead and resume the video here.
And we're going to have a single paired end read. We just have one absolute file path. So I'm going to Control X to exit out of that. I'm going to zoom in a little bit because I like to be able to see what I'm typing. And we are going to actually import the data. So we're going to scroll down to this QIIME tools import. We're going to copy from this little guy, and we're going to paste them in. So the QIIME tools import has a type, which is the sample data with sequences. This basically indicates that we have sample data that was single end reads and has quality information. The input format file, basically it says that it's a single end FASFA manifest thread 33v2, which means that QIIME can expect a manifest file that has a sample ID and one absolute path. It will also assume that the Fred offset used for the positional quality score is in all of the files is about 33. For Fred 33 is pretty standard for modern Illumina runs. And then finally, our input path is a manifest.qzv file, a manifest.tsv file, and we tell QIIME where that is, and then we tell QIIME that we want it to output our dmux.qza file. So now that is all imported, so let's ls, and we can see that dmux.seek.qza file. So that's awesome. We have data. It's imported. Beautiful. Now we need to look at our data. So how we need to do that is we need to run QIIME dmux summarize in order to get a visualization. So let's look more into that little command. So most QIIME commands you can run --help in order to see more information about the command. So here we can see that we can input data. It has to be an artifact. We already have that. You can see the sample data quality with sequences with quality probably should look pretty familiar. The paired end sequences with quality and joined end sequences with quality are also inputs that this command takes, but our data is sample data sequences with quality. This parameter says how many sequences will be selected for the quality plot scores. We're not really going to mess with that. Our output says like where our visualization will be and what it will be named. And then there's some miscellaneous stuff like output directory, which outputs unspecific results to a specific directory, verbose, which makes the output like more wordy, like it explains more what you've done, and then quiet, which means it says nothing if it's run perfectly. Then there are examples, citations, and also help for the command, which we just ran, right? But now that we've taken a look into QIIME 2, or not QIIME 2, now that we've taken a look into QIIME dmux summarize, let's actually run that command. So as I said, we have an input of our data that we ran dmux seeks.qza, and then a visualization called dmux seek.qzv. So I am running that, and that should visualize our dmux seek.qza into a visualization. So as you can see, we have our visualization. So ls, and we can see our visualization in our directory. Awesome. So now we want to visualize it. How we're going to visualize things in this workshop is we're going to go to workshop-server.qiime2/zippyowl. Zippyowl is mine. You enter your username. Then we're going to go to the mouse tutorial, and we're going to right-click on this dmux seek.qzv file. We're going to copy link address. Then we're going to head over to QIIME 2.
All right, we're going to pause that real quick and just give everyone a chance to catch up. I am going to send out another poll really quick. Whoops, I used the wrong list. So if you can go ahead and let us know where you're at with the steps in this importing section tutorial, that would be really helpful. Let's see, I'm looking at the question thread here. Looks like perhaps there were some copy and paste issues, Aurora. Maybe you'll want to, let's see, you've downloaded the sequences and you have the manifest file. So I think the next step there is going to be copying and pasting the import command again, where it says QIIME tools import. Let's see. So Aurora, you'll want to rerun the QIIME tools import command. Let's see, where else are folks at? A lot of folks are all done here. The server address that we need to go to view the results. So if we can turn on my screen share, that would be really helpful. We'll show this. So Evan just shared. So the URL to look at the results is view.qiime2.org. And then the URL that you'll want to open to look at your, or to actually get the URL for your results is workshop-server.qiime2.org/your username. So my username is Astute Salamander. And this information will be on your PDF cheat sheet as well. And like Evan pointed out, this will be covered again here in the video. We just wanted to pause really quick, make sure that everyone has a chance to catch up. And so if you are running into an issue with importing or with creating and viewing the demultiplex.qzv, we will have instructors send you a private message to initiate a debugging chat here. So make sure that you keep track of those messages and notifications, just so that you don't miss that. Let me see here. So if you were stuck before and got moving forward, please update the poll. So I think you can deselect your previous option and then select a more appropriate one. So that would be really helpful for our instruction team. All right. I think we will go ahead and resume this video right now then.
Then we're going to go to the mouse tutorial, and we're going to right click on this demuxseq.qzv file. We're going to copy link address. Then we're going to head over to QIIME 2. View, my favorite website on the internet. And we're going to view our results. Typed it wrong. Don't know what I typed wrong, but Google figured it out.
After we've gotten to QIIME 2 view, let's click on that highlighted link from web.
Okay. And we're going to go. And so QIIME 2 is going to be able to visualize it. Let's walk through a little bit of what we see here. So here's our sequence count summary. It basically says what the minimum count is, what the mean count is, what the maximum count is, and what the total amount of sequence counts in our data is. We can kind of see that visualized a little bit in this forward read histogram, which allows us to see that the majority of our samples have sequence counts that fall between 4,000 and 6,000. And some of our sequence counts, no, not some of our, one of our sequence counts is 16,000. And then you can see that in more detail. I'm going to zoom in just a little bit. In more detail, here where you can see the sample ID and the amount of sequence counts for that sample ID. So for instance, this sample has the 16,000 sequence counts, while like this sample has 5,408 sequence counts. So awesome. Then we will go to our quality interactive plot. So this basically allows us to see the quality of our plot. I'm going to zoom out just a little bit so that I can get this plot and my chart in there. So basically what this plot lets us do is it will allow us to see the quality of our data at a specific position or a specific grasp of positions or just in general. So right now we can see that our data is pretty good all over. If we scroll in to just see a couple of them, we can see like points 4 through 19 or 20, which is awesome. And then we can like look at a specific one and get more information on its quality scores. So for position 13, we can see that its average or its median quality score is 37, which is great. And its highest one is 38, and its lowest one is 17 in this plot. Awesome. So that's definitely like information that's very helpful. And then a little bit further down, we can see the sequence length summary, which just says how many nucleotides are in each sequence, which is 150. So then we're going to go to Providence. So Providence is awesome because it kind of keeps track, a record of whatever you've done. Not very descriptive here because all we've done is import our data and visualize it, but it is something that we're able to do. So right here we can see the square kind of tells us like the actions we've done. So right here we can see that we have imported our manifest, like our data through a manifest. Here we can see that we visualized our data using the action summarize. And then these little dots is kind of the result details. So it explains the results that we got. So this is, we got our imported data. And this says that our result was a visualization. Awesome. It's great to like have that information for the future more like it. So if you go back to this, if you go back to the data you've run analysis on and you go like, I have no idea what I did, it's awesome because QIIME has a Providence. But now let's go back to our QIIME 2 view and answer some of those discussion questions. So I'm going to skip down to the bottom one real quick, and it says, if you're working on the tutorial alongside someone else, why does your plot look slightly different from your neighbors? If you aren't working alongside someone else, try running the commands a few times and comparing results. So the answer to this is actually right here. These plots were generated using random sampling of 10,000 out of 250,000 sequences without replacement. So basically what this means is that everyone gets a slightly different quality plot because it's randomly generated out of 250,000 samples and you only get 10,000 of those samples. So although we don't have the same sample, like the quality plot, they should be roughly the same because hopefully everyone has a representative 10,000 samples. Okay, so I'm going to go up to the top of the discussion questions now, and it says, after demultiplexing, which sample has the lowest sequence depth? So we can find that in our overview of our demux file, and we're going to find that in our per sample sequence counts. So this allows us to look at a sample and see what its sequence counts are. So this is the highest sequence count, so the lowest one's going to be at the bottom. So our lowest one is this guy, recipient for 460 W, I'm not going to say the whole thing, but this guy down here. And then its sequence counts are 4,237. Awesome. Now, what is the median sequence length? So we can find that again on the interactive quality plot page, where the demux sequence length is 100, like all of them are 150 nucleotides. So the median length is 150. This is pretty common to have all of them be 150 for Illumina runs, but it may differ if you use different sequencing techniques. Just to clarify that, that was a little confusing. It's common for Illumina runs to have all of the same length. Ours are specifically 150 nucleotides. And then our next one is, what is the median quality score at 127? So 125. I don't know where I got 127. So I'm just going to zoom. That's not helpful. To back out of a zoom, you double click on the graph. So I'm going to zoom into a little bit of this so that I could just get a I can get a better view of 125. So this is sequence 125. And if we look at our plot, we can see that sequence 125 has a median of 38. So now we're going to go on to our checkpoint. What is a good position to consider trimming or truncating at? So trimming and truncating are for denoising, and we'll get into that later in our next section of our tutorial. So I'm not going to go too much into it, but we are able to see that our data is pretty even overall. So that gives me an idea that we might be able to use the full 150 base pairs. But again, we'll go into that in more detail in the next section. So what did we learn today? We learned how to import data, how to visualize the dmux.qza file, and how to read it, as well as start to understand where might be a good place to trim and truncate our data. Thank you guys so much for listening, and I hope this was helpful.
All right, welcome back. Thank you, everyone, for letting us know where you were at during that sequence of commands that we were running in there. And we hope that everyone has gotten a chance to get caught up. I will mention really quick, Aurora, Chris Keefe is trying to initiate a video chat with you in your private messages. So please check your private messages, and he is standing by waiting to lend a hand. So we have time for maybe one or two quick questions before we head into our first break of the day. So Bod, we had a question come in about the meaning of that FRED33 v2 part of our import command. So the question is, does that FRED33 v2 mean that this command will only import sequences with score 33 or higher? Would you be interested in answering that?
Sure, I can certainly give it a go. I can't remember from the other videos. I think maybe Andrew covered this in one of his sections. Maybe it hasn't been done yet or not, but or I could just be imagining things. Essentially, the 33 corresponds to the ASCII scores that these quality scores start. And so there are two different versions of this, an older version, which was the FRED 64s, and the new version, which is FRED 33. And the important thing here is to just recognize that QIIME2 isn't able to automatically detect which of these file formats you have, whether the older one, which would be 64, or the newer ones, the FRED 33, which is most common sequencing files that you would work with these days. So that's really a parameter just to tell instructing QIIME2 that this is the file, the FASTQ file format that I have, and that the interpretation of the ASCII letter should correspond with that, the instructions specific for that file format. I'm sure there are probably a little bit better ways to describe that, but hopefully that gives you an idea.
That's that's totally it. And again, this is a really good question for a sequencing center as well, if you're ask them. But like Bod just mentioned, basically any recent sequencing product is going to be this FRED 33. It's really a big question with older sequencing data. And yeah, like Bod said, part of the issue here is we actually can't automatically detect which score scheme you might be using, because there's overlap between the ranges of score values here. So you can't necessarily say for sure, at least programmatically. So I have a question coming in from the control room about that V2 part. So in QIIME2, we have these semantic types and then also data formats. I covered a little bit about this yesterday in the morning. And this format here is specifically a modification of an older version of the FRED 33 format. So we had a FRED 33 import format that used, I think, CSV files. And at some point we realized, maybe we should improve this format a little bit. So we made a new format and gave it an all new name, was very original. We just put V2 at the end of it, and that's it. So this is how you kind of instruct QIIME 2 and provide that context to QIIME 2 about what the data there means. Have another great question here, Bod. What quality score is considered acceptable? This deals with trimming that will do, correct?
So, yeah, that's a really good question, and I don't think I want to give a definite answer because I don't think there is one. But like I mentioned before, these are probability scores. And in the old days when we were using OTU clustering and we were using, this is a question that might actually be a little bit more clear after the next section on denoising. But essentially, when we were doing quality filtering in the past, we were basically dropping values that had a quality score or Phred score of 20 or less, and that was sort of the field minimum standard. Of course, people recommend that you always use higher ones, but that was sort of what was accepted. But after the denoising video, you should have a better idea of why those are not necessarily as important as they once used to be, given the new quality control algorithms that uses to correct poor reads and so on. I don't have a good recommendation cutoff. Of course, highest is better, but that also really depends on how your data needs to be used. If you drop too many reads, then you might have proper merging of your reads. So there's a lot of other considerations, but I do want to maybe revisit this question after the denoising to see if that still isn't clear.
Cool, thank you. So we ate a couple of minutes into our break here, so I won't hold you any longer. We will meet back at the start of the next hour, so that'll be 11 a.m. Mountain Time. So thank you to all of our instructors that gave Q&A and presentations and everything this morning. And we'll see you all in 23 minutes. All right, welcome back, everyone. Hope you had a good break. Sorry we cut it a little bit short there. We just wanted to make sure we got through all those great questions. So in this next time block here, we have three different segments that we're going to go through, and all three of these have quite a lot of time baked in for Q&A and discussion and hands-on work as well. So just kind of preemptively letting you know, we've got plenty of time baked into this next 90-minute section. So Bod is going to kick us off with a walkthrough on some theory for denoising methods and how those relate or don't relate to OTU clustering and some historical aspects. And then after that, Chloe is going to walk us through the next steps of the Parkinson's mouse tutorial and actually apply one of these denoising algorithms to the sequencing data that you just imported. So with that, we're going to queue up Bod's video and send your questions our way. Thank you.
Hello again, everyone. My name is Merbod Astaki. I'm a postdoctoral researcher in the Night Lab here at the University of California, San Diego. And in this video, I'm going to give an overview of the denoising and or clustering processes involved in marker gene sequencing data within the QIIME2 environment, and show you what is involved in creating a feature table that will be used as the basis of just about every downstream analysis you do in QIIME2. In the last lecture video, we went over the process of importing your raw sequences into QIIME2 as well as demultiplexing them as needed. So in this lecture, we're going to discuss the next step, which is the denoising and or clustering process that also incorporate a series of actions ranging from quality control filtering, trimming, resolving ambiguous nucleotides, dereplication of reads, and ultimately ending up with a feature table as well as its corresponding representative sequence files. So picking up where we left off from the last video, after importing and demultiplexing our sequences, here we have a sample data artifact which contains a series of per sample FASTQ files. Following the denoising and or clustering step, which we will discuss in more detail in a bit, we'll be provided with two new very important artifacts. The first one is a feature table artifact, and here I'm going to use the term feature to refer to any sequence, any unique series of sequences that was processed following the denoising step. You may have heard these tables also referred to as OTU tables, taxa tables, species abundance tables, or something else completely. But in this series, I'm going to refer to these as feature tables, again for reasons that I'll describe in just a little bit later. This feature table is essentially a summary of all of our sequences organized as a sample by feature table, and the values in the table represent the total frequency by which a given feature was observed in each sample. So for example, feature 1 was observed a total of 42 times in the orange sample. It was found 12 times in the blue sample, 25 times in the red sample, and so on. The second important output artifact we get from this process is a set of representative sequences. This representative sequence file, which is of the type feature data sequence, is essentially a list of the unique features found across our entire dataset and their corresponding DNA sequences. Unlike the feature table, this artifact does not hold any information about the actual frequency of these features or their association with any particular sample. This file is used in downstream processes, for example when we want to construct a phylogenetic tree for our dataset, or assign taxonomic classification to our features. So up till now, we've been frequently using the term denoising, clustering, and features. But what exactly are these and why should you care? So to answer these, let's start with a simple example. Here we have a hypothetical unknown sample that contains several species of bacteria, here denoted by the different color circles. These species are found in various abundances as indicated by the size of their circle. So for example, the green species here is found in very high abundance in our sample, while the red species is found in very low abundance. Now in reality, we don't know the true composition of our samples, so we plan an experiment to sequence this sample to try and figure out perhaps what is inside of it. So as before, we extract the DNA and we perform a PCR to amplify our marker gene of interest. During this PCR process, however, several things can and often do happen that introduces some level of deviation away from the true community composition. This can happen, for example, from PCR error, where small mistakes are made during the copying of the sequences by the polymerase. Perhaps a nucleotide is copied incorrectly, or an insertion or deletion occurs, which introduces a new artificial sequence that is different from the parent sequence. We can get formation of chimeras, where two unrelated sequences are combined, or we simply miss a very rare taxa that was present in the original community, but we simply didn't have enough depths or PCR material to capture the rare taxa. In all these scenarios, a certain level of noise is introduced that blurs the original community composition. During the sequencing process, there is also additional noise introduced to our sample in the form of sequencing errors. For example, if a C nucleotide is accidentally read as a G, sometimes barcodes can jump across samples, or even some amplicons from other cells or runs leak in, introducing contaminated sequences, albeit in very low abundances, but nevertheless they were never found in our original sample. So how do we deal with this noise in our data? Well, in the past, strategies to reduce this noise often involved a series of quality control steps, such as filtering reads with poor quality scores, trimming off the poor quality tails of the sequences, identifying and removing chimeras, or using curated reference databases as a basis of accepting true sequences. And finally, the remaining sequences following these quality control steps were clustered into OTUs, or operational taxonomic units, based on some sequence similarity threshold. Most commonly 97% was used. One of the downsides of this approach is that two distinct but similar species, in this case take the blue and purple species, could ultimately be collapsed into a single cluster. These clusters can also contain a variety of spurious sequences, as we discussed before, that were introduced as noise during the sequencing process. This also leads to the question of which of these sequences within each cluster should be used as a representative of that cluster for creating phylogenetic trees, for example, or taxonomic classification, and so on. There are still numerous clustering algorithms being used today, all with their own strengths and weaknesses. Most recently, however, there has been some significant development in the field with the introduction of so-called denoising algorithms that do a substantially better job of minimizing the noise introduced between our sample collection and sequencing steps. These denoisers attempt to model the error profile of our sequences based on their quality scores, the expected error rates, as well as the observed frequency of each of these unique sequences. And so it uses these to model and ultimately resolves, or attempts to resolve anyways, errors in our sequences. Using these approaches circumvent the need for clustering your sequences, and so what you are left with is a high resolution analog of those traditional OTUs. You can think of them as essentially 100% similarity OTUs, whereby even a single nucleotide difference between two sequences can be identified with good confidence. So importantly, these denoising methods, unlike most other OTU clustering methods, are also highly parallelizable, which makes them perfect for projects with large sample sizes, which is increasingly prevalent in the microbiome field today. So what are the products of these denoisers called? Well, unlike the product of clustering methods, there isn't a single standardized name, and the developers of each of these tools have their own preferred terminology to refer to these exact sequence variants, or the features. For example, the developers of the Data2 tool refer to their products as amplicon sequence variants, or ASVs. The authors of the Blur tool refer to theirs as sub-OTUs, or SOTUs. Developers of the MET tool from the Marine Lab also refer to their products as ASVs. And finally, Robert Edgar, the developer of the UNOIS3 method, refers to his products, the product of his algorithm, as zero radius OTUs, or ZOTUs. However you want to describe your data is up to you ultimately, but in the QIIME terminology, we simply refer to these as futures. That is because these terms, at the time of their conception, were created really with a focus on using them with target gene amplicon data, while QIIME2 is agnostic to the type of omics data used, as long as they can satisfy the requirement for their semantic type. This also allows the same infrastructure that here is used for 16S target gene data to be used for any other data type, any other type of omics data, such as long read marker gene data, shotgun metagenomics data, proteins or metabolomics data, and so on. So you will likely hear the term future most frequently used within the QIIME2 environment. And while I personally think that the product of these denoising methods, so these futures, are a superior way of looking at our data, this is not universally accepted. I know a few prominent members of the microbiome community that still prefer to use OTU clustering methods in some circumstances, and have made some interesting arguments against the use of exact sequence variants when it comes to short target gene sequences. Now, whether these exact sequence variants or OTUs should be used is worth its own lengthy discussion, and is beyond the scope of this lecture. But I will say that there may be some scenarios where clustering your data can be beneficial to a specific biological question you may want to ask of your data. For example, in this example data set, we have two distinct green features in our sample and three distinct red features that are found in various frequencies. And say that the two green features are bacteria capable of digesting plant materials, whereas the red features represent bacterial clades that can better digest meat. And perhaps your biological question is related to the composition of plant versus meat digesters in various gut communities. In this scenario, clustering these closely related species may increase the probability of identifying statistically significant differences between your treatment groups, largely due to the increase in power obtained by clustering the green features together and the red features together. So in this particular scenario, OTU clustering may be, and again, this is always up for debate, but it may be desirable. However, even in rare scenarios such as this where OTU clustering may ultimately be desired, I would recommend that still the starting point should be with denoising your data and clustering the output of that into clusters if needed. So yes, you can do both. You can denoise and cluster your data. The advantage of this approach is that we can still use our denoising tools, which have by far superior quality control methods implemented in them, to produce the highest possible resolution of our data, and then cluster our features down to whatever similarity threshold that we want. Of course, you can always just use traditional OTU clustering methods without denoising first. And in fact, if you have old data types without quality scores, this may be your only option. But in general terms, we advise that as a starting point to use the denoisers, and then the choice of clustering or not clustering should be strictly driven by your biological question, and this should be a user-driven choice. So this concludes this lecture video on denoising / clustering methods in QIIME 2. In the next tutorial session, you will get to learn how to perform denoising on your actual reads using QIIME 2, the Data 2 plugin. So thank you for joining, hope you enjoyed it, and see you next time.
All right, welcome back, everyone. I'm joined in the control room by Bod. Thanks for that walkthrough there on denoising. I've got a question for you here. During your lecture from L. Brian Patrick, in case it's not covered, struggle to determine truncation values. I see in the tutorial that the full 150 nucleotide sequence is used, but this seems highly subjective. I'd be interested to know some more objective criteria for determining truncation.
All right. That is a wonderful, wonderful question, and probably one of the most common questions we get on the QIIME 2 forum is, how do I select those trimming, truncating parameters? Now, in rereading this question, Brian asks more of an objective criteria for determining this, and I'll answer this in two different ways. One, that objectively in this case might not always be the right way, because your choices of trimming and truncating will really depend on what the goal of your data analysis and some of the requirements that it's going to cater to that specific data set. I'll talk about that in a little bit. But also, if you still insist on having a more objective way of doing this, there are at least one tool I know that tries to use an optimization solution for this, and this is a tool called Figaro. I can provide a link in it later. It's not a QIIME 2 plugin yet, as far as I know, although the developer of that tool has sort of expressed interest in creating a QIIME 2 plugin for it. So it might be down the line something we will have access to. Now, so the question really comes down to, how do I select these trim and truncating parameters? And like I said, we've done a lot of example cases on the QIIME 2 forum. So if what I say here doesn't seem to satisfy it, please feel free to search through the forum, see how we get exact values and numbers, and then you can chime in there. So, Matt, am I okay with time if I go a little bit deep into this? Yeah, okay. So, two things to consider. One, is your data single-end read or is it paired-end read? In single-end reads, we are a little bit easier. We can select these parameters a little bit easier because we don't have to worry about our reads needing to merge. So the forward and reverse reads don't need to merge. And for people who, I just realized maybe they're not familiar with what we're talking about when we talk about trim and truncating parameters, is when we are performing these quality control, such as the denoisers we talked about, one of the first steps in those algorithms is to try and remove low quality, so those are the Phred score, low quality tails of our sequences. The reason why we want to do that is because those really poor quality, so if the Phred scores are really low, those don't provide any useful information, and in fact, they may end up forcing us to call a wrong nucleotide, which can, disrupt the true signal that we're trying to find. So one of the first steps in those processes is to get rid of those poor quality tails. And so in essence, if you have 300 nucleotides, but in Illumina sequences, the last 100 or so nucleotides actually drop vastly in quality. So oftentimes it's better to get rid of those than keeping them and, garbage in, garbage out, essentially. So if your input data is not good, then the output is also going to be unreliable. So this is sort of what we're discussing here, is those poor quality. Now, with single-end reads, like I said, it's a little bit easier because you don't have to worry about your reads merging. So you can look at your quality score bar plots that we showed in an earlier video and see where that quality score starts to drop. And you can make a decision based on that alone. If you think your nucleotide sequences at 100 or 150 nucleotides are good quality, you can trim it there. If you do happen to have 300 nucleotide long reads using the V3 chemistry kits, or is it V4? I can't remember. The most you can get out of Illumina reads, then in those cases, yes, you do get a higher resolution when it comes to your taxonomy, but the quality score might be low. So there's a trade-off there. Again, into your consideration of your question, is resolution really important or is quality important in your community analysis? Now, when you're considering pair-to-end reads, then there's another dimension to this consideration, and that is using the existing Data2 plugin. I'll use that example. It requires that your forward and reverse reads overlap by at least a minimum of 12 nucleotides. So a more concrete example, in our V3, V4 primers, which is a very common primer we see in the literature, there's about 140 or 150 overlap between our forward and reverse reads. And so when we're making those decisions about truncating, so removing from the 3' read, we need to account for that we can't truncate too much, otherwise our reads won't overlap. So when you're doing your calculation, you consider how long your total sequence length is. So your forward and reverse together, each are 300 base pairs long. That adds up to 600. We have about 150, I'm using this as an example, 150 nucleotides overlap in the middle. So we have to make sure that whatever we truncate from both sides does not surpass 12 nucleotides. So that's the bare minimum we need to keep to make sure that they can overlap properly. So within that realm, there's a lot of other fine-tuning you can do, but those are the two major considerations. So the more of your poor quality tails you remove, the better Data2 will perform and the better quality your reads will be. But if you go too far, then you'll actually start losing reads altogether because they aren't able to merge. And if they don't merge, tools like Data2 will just discard those altogether. So there's this sort of optimization to do, and you can do that manually or using Figaro if you want to play around with that. That gives you an ideal situation. So in some cases, you may consider keeping these high quality sort of only and get rid of some reads, that might be okay with you. Your coverage was good, so you can afford to lose some sequences. But in some cases, maybe your sequencing run wasn't all that great, so you can afford to lose those, and you need to be a little bit more lenient and allow poorer quality tails to keep sequence. So, okay, this is a little bit of an overview, and I know it's probably got a lot of you confused, but it's definitely something that you will interact or you will run into in the future if you're dealing with paired noise. And when you do, you can come and chat with us on the QIIME2 forum, and we'll do this again and go in much more detail. I think I'll leave it there for now.
Cool, thanks, Bod. So we have a couple of questions came in related to OTU clustering in particular. Control room, I'm going to share my screen really quick. So we aren't going to cover OTU clustering specifically in our tutorial. In the Parkinson's mouse disease tutorial, we're going to be using Data2 to denoise our sequences. But I just wanted to kind of point out how you could go about finding information about how to perform OTU clustering in QIIME2. So I have opened up here in my browser docs.qiime2.org, and I'm looking at the latest version of those docs. And if I scroll down, there's a whole list of tutorials here. We've been looking at the Parkinson's mouse tutorial. I'll direct you down to the clustering sequences into OTUs using Q2V search. And if I can get one of the instructors to drop a link to this in the discussion topic, that would be really helpful. And so this tutorial, it's a shorter tutorial than the Parkinson's mouse one, but this has explanations of performing a few different types of OTU clustering. I will not go through all of the specifics here, but this would be the kind of thing that if you decided to work through and had questions about, you could come visit us on the QIIME2 forum, or if you work on this week, you could ask in one of the discussion threads some more specific questions. There was also an OTU clustering question specifically about how do I change the similarity threshold for OTU clustering, say if we were doing de novo clustering. And so I'll just point out here in this tutorial, it actually includes an example of a 99% threshold for de novo clustering. And so this parameter here is where you would go ahead and change that value. So if you wanted to set it to 70% or 85% or something, you could change that. I'm going to turn off my screen share now. And Bod, there was another OTU clustering question about is it acceptable to use 98% or 99% similarity as opposed to 97%? Do you have any thoughts on OTU clustering threshold?
Yeah, that's an excellent question as well. And unfortunately, there isn't a very clear answer to that either, as with anything we've been hearing, that it depends on what you want to do and the data itself. 97% was historically chosen based on this idea that at 97% similarity of DNA sequences, you can differentiate between species. Although that particular threshold has definitely been challenged, and I know a lot of people who prefer to just use the highest resolution possible without clustering. Most of us, I think, at least default to ASVs or the 100% OTU, if you want to think about them that way. And under very strict and specific considerations, we might do OTU clustering. As to what if you are deciding to do OTU clustering and what would be the appropriate percentage, I can't really give you any recommendation there. I mean, 97 was the de facto standard, but I know some people like Pat Schloss, the creator of Mother, he's actually got a series of videos that he goes over this exact same topic on YouTube. And his most recent suggestion was that perhaps that threshold should be determined on the fly and be driven by your data. And so, maybe 97 is not always the right thing. Maybe in some cases it's a little bit more, sometimes it's a little bit less. So, yeah, there's a lot to consider. And unfortunately, it's not this very clear sort of answer on what to pick. Again, for those reasons, we tend to stick with the ASVs. They have several advantages over the OTUs. And if you were to use 97 though and publish or submit a paper with that, I don't think anyone would question why you chose 97 over 98 or 96. So with that regards, you have some, at least there's some sort of solace there that you won't have to worry about justifying it. But if you really want to get down to the nitty gritty of it, it might be that you need to actually determine that threshold based on your data.
Yeah, and things get complicated too with like meta-analyses, for example, or if you're, looking to continue an older established study and following on with that protocol. Let's see, we've got time for one more quick question. For paired-end reads, would it be okay to trim the forward and reverse reads separately, or do the reads need to be joined first before trimming?
Yeah, I think Mike may have actually answered this already in line in there, but basically it depends on which tool you're using. So when you're using a tool like Data2, it does process your forward and reverse reads separately first, and then it tries to merge them. So you would actually have to provide it with specific instructions to remove those, truncates from forward and reverse separately. But the other denoising algorithm we have in core QIIME2, deblur, really only deals with one direction of your reads. So by default, the forward. It still does accept paired-end reads, but you do have to do that joining yourself manually before loading it too. So we do have a plugin in QIIME2 to do to join paired-end reads. So if you are using deblur, you'll have to do that before, with Data2 during. So it just depends on which approach you're taking.
Cool, thank you. So we are going to put some of this theory to practice in the next section. So Chloe is going to walk us through the process of denoising the sequences that we imported in the last section of the tutorial. So if you are not logged to Workshop server right now, would be a really good time to do that. Make sure you have your PDF cheat sheet open, and then I'm going to share my screen really quick here and show you where we're going to pick up in the tutorial. So you'll want to have that tutorial open, and in that last section we covered QIIME DMUX summarize, and we looked at the summary of our imported data. And so then this next section starts here with sequence, quality control, and feature table. And Chloe is going to walk us through these commands and these next steps. So with that, take it away, Chloe.
Hi all, in this section of the tutorial we are going to perform some sequence quality control and denoise our data. First, we will run QIIME Data2 denoise single right here, and then we will run QIIME metadata tabulate. So before we get on with the fun of this tutorial, let's talk about where we came from, what our last tutorial was. So in our last tutorial, we had imported the QIIME2 data. And at the very end of our last tutorial, we were looking at the dmuxseq.qzv file, and we were kind of like talking about what it showed us. So the first step of denoising is actually figuring out where to truncate using that visualization. So let's make sure that everyone has that. So first I'm just going to ls in my working directory of the mouse tutorial. And you should see this QZV visualization. And if you don't, then maybe you should return to the importing and demuxing part of the tutorial and then come back to this video because that's a pretty important part of this tutorial. So before we jump in
to actually doing some quality sequence control and denoising our data. Why do we need to do this? So we need to denoise our data because there's noise in general in our sequences that come from sequencing and sample collection. And this could affect our analysis or like how we view the composition of whatever we're looking at. So we need to make sure that the sequences that we're looking at are representative of what we're trying to analyze. So in this section of the tutorial, we will look at where it's appropriate to truncate our data, how to denoise our data, and how to interpret a data2 stats.qzv file. And at the end of this tutorial, we should have good quality data that doesn't have too much noise that allows us to move forward with the QIIME2 pipeline. So first, we need to look at this visualization in order to kind of see what the quality of our sequence is and where we theoretically should truncate. So we need to go to the location where we can get the visualization. So we're going to go to workshop-server.qiime2.org/zippyowl, which is my username. So make sure to enter your username. So I go to my mouse tutorial directory, and here you can see all of the files that we could see over here, just like slightly differently, just organized differently, visualized differently. So I'm going to go to my QZV file. We have one called the same thing, that's a QZA file, so make sure you go to your QZV file and copy that link address. Then I'm going to go to QIIME2 view, and boom, bada bing, bada boom. We're going to provide a link from the web. And we're just going to paste that little guy in there. So looking at this, it tells a lot about our DMUX sequence summaries, but we want the interactive quality plot. So I'm going to zoom in just a little bit here so we can see this plot a little bit closer up. Maybe that's a little too much. Okay. So here's our plot on our forward reads. This is a single end read, so we don't have a reverse read. And we can kind of see that our data is pretty good. There's no real hard and fast rule for how you should truncate your data. It's mostly a judgment call, but it has to be a rough estimate of the good data. Sorry, what I'm trying to say is it has to be, relatively good data so that data 2 doesn't get lost in all of the noise and the bad quality and stuff. So a good estimate is probably like a median above Q30. We can see by the box plots, I'll zoom in. You can see that by the box plots here that there are the fair amount of our data has a median above Q30. So that's really good. There's no major drop off around here, which typically happens with data. So that's really good. Since our data is overall so good, we don't really need to truncate at any specific length. So we will include all of this sequence length in our analysis. So we will truncate at 150 sequence bases. So let's go to this tutorial again. So here is our data2 denoise single. So now that we've decided where to truncate, we're going to run this. It takes a while to run, so as it is running, I'm going to discuss what we've inputted and what we'll output from it. But at first I'm just going to run it because it takes a minute to run. So we got it all in there, press enter. Okay, so first we inputted a dmux-seq.qza file. This is a zip file that contains each read. So when we denoise these reads, we'll still know which read belongs to what sample. We didn't use the parameter trim left, but it is an option for QIIME data2 denoise. And what it does is it trims out the primer if the sequence still has the primer in it. But our data is EMP protocol, which means that it doesn't have the primer still in it, so we didn't have to trim left. We will output a table which has sample IDs and sample features, but has no interest in sequences. So this table right here will have sample IDs and numbers of features, but it won't have any sequence information. This representative sequence file, believe it or not, has sequences. It's an output of the amplicon-seq variance, and all of the corrected sequences are given a number called a hash. So whenever the same sequences are found, it produces the same hash. This is a way it's easier to refer to the same sequences because that hash is so much shorter than referring to them as like their entire name. And then lastly, we will output denoising stats data2 underscore stats.qza, which basically tells us how the denoising step went. As you can see by our next step, we're going to do QIIME metadata tabulate on that data2 underscore stats.qza. So that file is actually a metadata file. So we'll use metadata tabulate to visualize it, and it can be used later on in the pipeline as a metadata file if you wanted to do that. So we just got all of the outputs that I talked about. You can see that these are feature tables, and this is sample data. So the next step we're going to do is we are going to run metadata tabulate. So, bajam. We are going to run this, and then we'll get a visualization of that stats.qza file. So it didn't take very long, and now we have that stats.qzv file. Awesome. So now let's head back to that visualization of the workshop server. So basically, I'm going to refresh because this page doesn't auto-refresh when you get new sequences. So until I refreshed, we didn't have my new sequences that I had just created. So we are going to want this data2 underscore stats.qzv. So I'm going to copy this address link. I'm going to go to QIIME2 view. And I'm going to get a file from the web. So then I'm going to paste that in there, and it's going to create a create the visualization for me. We're going to be able to view it. So let's discuss what this is saying. So these are all our sample IDs, our unique sample IDs, and then these are all the reads from those sample IDs. So it's telling us how many were inputted, how many were kept after filtering, how many were kept after denoising, and how many were kept being considered non-chimeras. Chimeras are created during PCR when unrelated molecules become tangled and then they're amplified as a single unit. So for this first one, we inputted 4,460. Filtering based on quality, we kept 3,764. When we denoised that data, we removed all of that noise from the sequencing and sampling steps. We remained with 2879. And then when we removed all the chimeras, we had 2209. So pretty good. And then you can, obviously, like that's just for one sample. So there's a bunch of sample reads, and you can organize them by like lowest. So the lowest amount was actually had a huge input. It was the one that had our 16,000 input. We filtered out a lot of them, so we only kept 9,919. After we denoised them, we only got 347, and there wasn't any chimeras in there. They didn't filter out. I know it's kind of shocking to have that many reads filtered out, but the majority of our samples actually didn't lose very many reads, so we're not that concerned. It just means that that particular sample happened to be very noisy, and unfortunately it happened to have a high sequence depth. So there you have it, folks. We now know how to read a stats file from data2. So just to recap what we've done in this tutorial, we learned how to denoise our data. We learned how to do some quality control on our data. And just in terms of when to use this can be used a lot of times, whether or not it's data2 or another denoising and quality control method. This is a pretty important part of most pipelines because you need to make sure that your data is of good quality and doesn't have noise from sequencing. Anyway, I hope you guys enjoyed this video, and thank you guys for listening.
In this section, we are going to be generating a feature table summary. Now that we've effectively run data 2, we have our data 2 results. So we have our data 2
All right, welcome back. So I am just checking out the discussion thread here. Thanks so much for that, Chloe. So let's see. Okay, I'm getting some updates from the control room. So can I confirm, are we live right now here, control room? All right, so we are taking a quick break to just make sure that everyone is caught up, and then I think we're going to finish out this video to look at a feature table summary. Did I hear that right, control room? Okay. Okay, so let me just take a quick peek here. So yeah, there's a couple of really good questions about the interpreting the denoising results. So we'll hold off on that at the moment. Just want to make sure that everyone gets a chance to get caught up. Let's see. Yeah, it looks like maybe we can go ahead and finish out the video, and we'll lend a hand as people have questions here, and then we'll have some time for Q&A after.
Thank you guys for listening.
In this section, we are going to be generating a feature table summary. Now that we've effectively run data 2, we have our data 2 results. So we have our data 2 table.qza, our data 2 repset.qza, and our data 2 stats.qza. We have looked at our stats.qza, which allows us to see how our denoising went, and now we need to create a summary or a visualization for our data 2 feature table. The feature table includes counts associated with each sequence and each feature that will allow us to gain more insight on our data. So let's head on over to our command line and make sure that we have everything we need. So I'm going to ls into my working directory. My working directory is my workshop directory, and then inside that workshop directory is the mouse tutorial. So I'm going to ls, and what I really need to make sure I have is this data2.qza file. If I don't have that, I can't run this command. So now I'm going to come over and I'm going to copy from this little clipboard guy our command for running feature table summarize. And we're just going to paste that right in there. We're going to be running a feature table summarize. Our input is going to be the data2 table.qza. We're going to input our metadata, and we're going to output a visualization. And so this should only take a couple of seconds, and once we have that, we will have our visualization. So now we have our visualization. I'm not really going to look into this one, but we will look into it later on when we're doing our alpha rarefaction video. Awesome. So thank you guys for listening, and I hope you learned a lot.
All right, welcome back. Let's see. I'm going to pull up my chat here really quick. Let me see. So there was a question that came in earlier. Let me go ahead and get this pushed over to the control room. About interpreting our denoising, data2 denoising stats. So this question here was it's a little bit broad, but what is a good percentage that should remain after the filtering steps or the denoising steps or the chimera detection steps? And let me see, Bod, are you interested in answering that? or?
Yeah, go ahead. For sure.
Okay. Looks like Evan's going to join us.
Okay. Oh. So many buttons. All right, can you see me? Cool. So yeah, this is a really good question, and it does. Why am I still hearing myself? Because that didn't get muted. Okay, there we go. You know how when you have that like 5-second delay on your own voice, it's really hard to talk. So this is a really good question, and it comes up a lot. So kind of the benchmark, like the overall proportion is maybe less important than the proportion that are retained at any given step. So you don't want to lose the majority of your reads consistently at any one step of this process. That probably indicates something's a little bit wrong, either with your parameters or with the data. So a really common one is after like QIIME filtering, you lose like 80% of your reads. That probably means like you actually have your primers in your sequence data still. As just a very common example. So that's kind of what we're looking for. We don't want to lose a lot of reads at any given step, but if the total aggregate of all those steps loses a lot of reads, that's actually okay, because sequencing is a pretty noisy process. So if you lose half your reads over the whole thing, maybe that's not a big deal. If you lose half your reads after a single step, maybe that is a big deal. So it kind of is subjective, but that's what we look for. Hopefully that's helpful. Does anyone else have kind of thoughts?
Thanks for that, Evan. Bod, did you have anything you wanted to add to that, or?
No, I think that's pretty good. Highlighted the what to look for there. In my experience, when I'm dealing with V3, V4 like paired end reads, I tend to lose a little bit more just because there's a lot more fine tuning for sort of that parameter optimization. I tend to, on an average run, so consider all the highs and lows, I tend to keep about somewhere between 70-75% of my reads, but it is not uncommon to lose upwards of even 60% of 50 up to 50% of my reads, as Evan mentioned, throughout the whole process. It really depends on your sequence reads and not. So don't be shocked if you see drops of that much. It could be totally a good thing. You're just getting rid of bad quality stuff.
Another thing that's maybe worth mentioning is the author of Data 2, Ben Callahan, likes to mention that your goal isn't to find the best parameters. So don't stress out too much about finding the best possible recovery of reads. Your goal is to find good parameters. So Data 2 is a very adaptive algorithm and will it will find the signal in the noise. So as long as you're pointing it in roughly the right direction, it's going to give you the results you need. So don't worry too much about finding the perfect parameters. Like plus or minus one base is not really going to matter to the algorithm.
Thank you. Let's see. The next question that we have here is from Ryan. How does one know if the primers are still present and if trim left is needed? Chris, you available to take that one?
If my audio works, I sure am. Yes. I think this is just one of those great opportunities to contact your sequencing center. You should have a pretty good sense of what kind of protocol they use and what the data looks like that they are giving back to you. And if you're not sure, you can always ask. It's also possible to open up those files and actually look at the reads and see whether your primers are present. But that is probably more in-depth than you want to go. And so I would rely on the sequencing center whenever possible.
Totally. Yeah, thank you. Bod, did you have anything you want to add to that?
No, that sounds pretty good to me. Nothing to add there.
Okay. Let's see. We have another question that came in a little bit longer here. Do you suggest to perform quality control before running denoising? For example, using Trim-O-Matic to trim the paired end reads. Yeah. Bod, would you be interested in taking that, or or is that Evan jumping in there?
Yeah, I think Evan is excited for this one.
Cool. Evan, welcome.
Hello. Yeah, so this is another really good question. And it really does kind of depend on the amplicon you're working with. So for something like V4, like bacteria, probably just kind of setting your trunk length is sufficient. But maybe something like ITS, where it's very variable and really you would want to do something like trimming. In fact, something that's really important to do is to make sure your primers are removed, because Data 2 is building its error model based on biological sequence, and the primers really mess that up because they're all identical across all of your sequences. So something that can happen, especially with shorter reads, is you'll actually get the reverse complement of your reverse primer on your forward strand and the reverse complement of your forward primer on your reverse strands. And that's something that you might want to trim out with cut-adapt, especially if you're doing paired end ITS sequencing. Similarly, you may want to drop low-quality reads, and you might have variable length reads. If you're doing paired end sequencing, this is actually not super. This is okay, because what you're going to do is end up merging them anyways, so you'll have that overlap, and so you'll be talking about the same part of the amplicon. With single end reads, that variable length may become problematic because you're going to be generating ASVs which are at different taxonomic resolutions, basically. And so maybe you have two sequences that were the same sequence, but since we did this upstream trimming, you're going to, Data 2 doesn't basically know that you did that. And so it's going to see this slightly shorter, what it assumes is a biological read, and it will become its own ASV, even if maybe that was actually the same base call, right? So if we trimmed it by just one because the base call wasn't so good, then we're generating kind of this ASV out of thin air, when Data 2 would have merged those together because it would have recognized them as the same. So in general, you probably want to avoid doing any upstream processing outside of primer removal, which is super important. And let Data 2 kind of keep as much of the data as possible, because that's how it's going. It is the error that it uses to correct the error. And if you don't leave it any error, then it can't do anything. Would kind of be the long short of it. Yeah.
Cool. Thank you. Let's see, I have another question that came in here. In samples with high host or other non-specific amplicons, I've found lots of data to be lost in Data 2. I assume it is due to QIIME filtering, as the positive control sample run with the other samples showed good quality for the run. Is there a solution that can be used in the Data 2 side, or would another denoising method work better for this particular scenario? We're thinking this one over in the control room here.
Yeah, sorry, I was just rereading the question. I can maybe take a go at it while others come up with better answers than me.
Sure, thanks, Bod.
So the sample types that have a lot of host contamination are definitely problematic. I've definitely had to deal with them before in the past. So, for example, I used a lot of mouse intestinal tissues, which, at least with our extraction protocols, came with a ton, in some cases more than the actual microbial cells, DNA cells. So, yeah, I can definitely sympathize with that pain, and you do lose quite a bit of it in Data 2. But for the most part, that is, a very good thing, because otherwise, those sequences that have those on-target sequences that for some reason have made it through are going to add nothing but noise to your overall analysis. So yes, they will be detected and they will be removed, but that is a good thing. If you have a suspicion that maybe data 2 is falsely calling a lot of your reads as chimeras, you can play around with it with that parameter in data 2. I think it's called min fold over parent. I can't remember the exact parameter there, but that sort of gives you a little bit of flexibility in how you want to define chimeras. So sometimes by increasing that value, you're allowing a little bit more leniency, so data 2 is not discarding as much of those what it thinks are chimeras. But I would be careful in doing that because unless you know for a fact, unless you can, maybe blast those individual sequences that you're suspect of, you can blast them to see what they hit, whether they hit, host sequences or some uncultured bacteria. I would be a little bit hesitant playing around with that too much. Because like I said, ultimately what you want to do is do get rid of those host ones. One other step you can do to be sure is once you are finished your data 2 step, you can do what we call a negative filter. And this is something that the Blur does by default, which is essentially is taking all of your sequences and comparing it to a reference database. So in the case of the Blur, it uses green genes as default, and it says, okay, these are what bacterial or 16S reads should, in theory, look like, or at least reasonably look like this. And if I have a sequence that looks nothing like that, then discard it, because that's likely an untargeted sequence. So I find that also a good way to get rid of a lot of reads that have slipped through data 2. Maybe they were chimeric, maybe they were host cells, and they didn't get discarded. So that's also one option to get rid of them, and I've had good success with that. I think that's all I had to say on that.
Cool. Yeah, thank you for that, Bod. Evan, did you have something you wanted to add, or?
No, just for the next question.
Okay. So yeah, the next question here came in from Christopher. So if I understand correctly, no trimming for paired end data before data 2, right?
This is actually the one time you totally can trim, and it's okay. Because there's going to be a merge step. So what data 2 is going to do is it is going to, let me think about that. Yeah, no, this is when it's fine. So data 2 is going to denoise each of those individually, and say we did have that situation where one ASV was shorter because we trimmed it, and the next one was a little bit longer. Data 2 is going to make those two different ASVs, but we're going to merge them later. And so ideally our reverse end is going to clarify that position. If it can't, then it's too short and they won't merge, and we'll discard it. And so we won't have this kind of ASV inflation from our trimming. The thing with the paired end is you do need to make sure that you have enough overlap between your two reads. And this is where amplicons like V3, V4 can be a little dicey, because unless you have like 300 base pair sequencing on both sides and a good amount of high quality reads, you may struggle to actually get that overlap. A really classic problem is where people might have like 2x150, or 2x200, which is theoretically enough to cover that amplicon, but in practice the data is just not that good towards the tail end of these reads, and so we can't make that overlap happen. So when you have paired end, you can trim, but it's because you overlap, and that's done by the data 2 method automatically. In the case of single end, where you're only looking at one direction, then you really need these to be the same length, otherwise you have different taxonomic resolutions for your ASVs, and they're not quite as comparable.
Cool, thank you. We had a couple of other questions come up in the discussion related to filtering based on taxonomic annotations. And I'm going to go ahead and queue those up for either our end of the day Q&A today, or perhaps we'll cover that tomorrow as well, after we go through the taxonomic assignment steps of the Parkinson's mouse tutorial. But that's a really great question, so we will follow up on that. And I think it looks like that is everything in our queue right now. So I think we'll go ahead and get the next video going here. So we are doing this next step slightly out of order in terms of how we've been doing things so far. So here we're actually going to run the phylogenetic reconstruction step of the Parkinson's mouse tutorial before we do the lecture. And this is because this step can take a while, 20 or 30 minutes. So it's helpful to go ahead and have that queued up and running while we're breaking and watching the lecture video later. So if you're not logged into the workshop cluster, now would be the time to make sure that you're in the system. And we will be picking up right where we left off in the Parkinson's mouse tutorial. So thank you.
Hi all, and welcome to the section of the tutorial where we generate a phylogenetic tree for diversity analysis. In this section of the tutorial, we'll be running two main commands. The first one being we_get, where we'll get our reference tree from the internet, and the second one being QIIME fragment insert sep, which is where we insert our sequences into that reference tree. But I'll explain all of that a little bit later. Before we get into creating a phylogenetic tree, we are going to recap where our last tutorial left off. In our last tutorial, we denoised our data. So you'll see a bunch of data to underscore sep, or underscore anything data, which all came from our denoising step. So now that we have good quality data, we will be able to make a phylogenetic tree. For our phylogenetic tree, we'll be using the data to repset.qza. And once we have our phylogenetic tree, we will have completed the last step before we actually get to the heart of the analysis. So let's head over to our other site and talk about why we are doing this. So later on in a lot of QIIME 2 pipelines, we will need a tree for our diversity metrics. Examples of diversity metrics that need a phylogenetic tree are FAES index and weighted and unweighted unifrac. There are a couple of diversity metrics that don't need a phylogenetic tree. An example of those are Bray, Curtis, and Jacquard. And it's important to note that this phylogenetic tree isn't proving relationships or publish worthy. It's just helpful for our specific diversity metrics. There's a couple of ways to make a phylogenetic tree. In this tutorial, we're going to be using fragment insertion, which is down here, right? Basically what this does is it takes a reference tree right here, and then it takes our sequences that we give it, and it tries to align those sequences to the reference tree and then insert them based on that. Okay, so now that we have a little idea of what we're doing, why we're doing it, let's start by running the commands. So first, we need to run we_get. So I am going to copy from this little clipboard guy and head on over to my terminal, where I'm going to paste in that command and run it. So now we've officially downloaded that from the internet. So let's make sure that we have it. So as you can see, the sepref file is there. So we're good to go. Next, we need to run the QIIME fragment insertion sep. So I'm going to copy it from this little clipboard and again scroll on over to my terminal. Now I'm going to paste it in there. So we need to talk about what we're doing. So before you run it, we need to change this. So I'm deleting this little comment that says to update to a higher number if you can. If you are on the workshop tutorial server, you can upgrade this to 2 threads. If you are on your own server, you need to select based on the resources available to you.
So now that we have this, you guys can run this command, and then I'll talk about what our parameters are. So our first one, these are our sequences that we denoised and now we are using to create our phylogenetic tree. Our reference database is our reference tree that we are using to insert our sequences into. It's also some alignment data and some metadata that is relevant to the SEP command that allows it to perform the insertion. This is one of our outputs, which will output the tree that we need. And this is an output of the placements of the sequences in that tree. It's something to note that QIIME doesn't actually use that currently. And then lastly, our parameter threads, which allows us to run on multiple CPUs, which will make this step faster. So my command has finally run, and let's go take a look at these outputs in QIIME 2 view. Go to our workshop server visualization, workshop-server.qiime2.zippyowl, to look at my data. I'm going to step into my mouse tutorial, and I'm going to go down here to the tree.qza file. I'm going to right-click that and copy link address. Then I'm going to head over to QIIME 2 view. So something to know, okay, and then we're going to put a file from the web, and we're going to paste that pathway that we just copied from our workshop server. So something to note is that this is a QZA. So a QZA doesn't really create a visualization like we've seen in our previous tutorials. So this is just more information. But let's just take a look at that little provenance real quick. So it's cool because you can move them around. So I like to move it around so that I can visualize the path a little bit better. So this is where we imported our data. This is where we denoised our data. And this is where we created a phylogenetic tree. And this is where we downloaded our reference tree so that we could use it for this command. So as you can see, our little provenance graph is definitely growing, and it's kind of exciting to see it take shape. So besides looking at the QZA format, we can also look at more of the phylogenetic tree by going to the Interactive Tree of Life website. So what we're going to need to do is head back over to that workshop server thing, and we're needing to download that tree.qza. And then we go back over to the Tree of Life website, and we name our tree. I named it PD Mouse Tutorial. I'm going to choose a file from my desktop. Awesome. And now I'm going to upload this. It can take a couple of seconds to upload, but then we'll be able to see our Tree of Life that we created using QIIME fragment insertion step. So here it is. So again, this isn't a particularly informative tree because it's not really for us to interpret that much. It's for QIIME to interpret, but we can kind of see how our phylogenetic tree looks like. We can also look at it in like long form or normal form or unrooted. It just kind of gives us a look at what our phylogenetic tree looks like. Very cool. Okay, and with that, we have completed this section of the tutorial. So in this section of the tutorial, we learned how to make phylogenetic trees for QIIME's diversity metrics.
Making a phylogenetic tree is particularly helpful because it's used in most QIIME 2 pipelines. Thank you guys so much for listening, and I hope you learned a lot.
All right, welcome back. So hopefully now everyone has at least started running the phylogenetic reconstruction step here. Chloe mentioned in the chat here, it took about 20 or 30 minutes for it to complete for her when she ran it. So it's fine if it is still running for you. We've got a break coming up here, so you won't need any of the next steps prepared. So I'm just looking at my chat here really quick. So Julie asked, so we're supposed to just follow along with our explanation while the program is running. So yeah, we're going to do some Q&A now at this point. You're welcome to just keep your terminal up, your Secure Shell app up. Let the results keep processing here. Could take about 20 or 30 minutes, and then we actually won't use any of these results until we pick up the tutorial again tomorrow. So the rest of today will be dedicated to the lecture related to phylogenetic reconstruction, as well as general Q&A. So yeah, it'll be important to make sure that you wait until the command is completely finished. So let's see. So I have a really good question that came in here. And if Mike is around, I'd like to see if you could take a stab at answering it. So the question here is, why would we run phylogenetic reconstruction before we perform taxonomic assignment?
Okay. Am I in? Okay, good. So the workflow, the pipeline you use can vary depending on what you're doing. So the order at which we're doing the workshop, a lot of it is to logistics, right? Can we fit certain slots of our discussions in appropriate times to fit? That makes sense. So I personally often will do taxonomy assignments prior to phylogenetics, because sometimes I would like to remove things that I'm not interested in or may not be appropriate to put in the tree. So earlier there was a lot of questions about chloroplast, plastid sequences, mitochondrial sequences in your data. So obviously you want to remove those, but a lot of times you won't be able to remove those unless you've classified those sequences first. So I tend to do that as well. One thing that you'll probably hear a lot more this week, and I'll just reiterate it again here, is there's no one way to do your microbiome analysis, right? So you need to adapt your pipeline as you go and what makes sense given the questions you're asking. Hopefully that helps.
Thank you. Yeah, so there's a couple of questions trickling in here related to the databases used. So I'm gonna just make sure that we get the last one moved in here for you into questions broadcast. So yeah, there's a couple of questions here about reference databases, like using like what would we pick for ITS? Or, you know, should we be picking green genes versus Silva? So there's a couple of these related questions pulled up here, and you know, we can defer, you know, as much of this as you would like until after the lecture. But you know, if you wanted to maybe say some words about that now, that would be great. And it looks like Evan's also in the chat, might have something to say.
Yeah, I just have something to add to that last point. So we haven't had and hopefully my audio is working. We haven't had the lecture yet, but as we go through Mike's lecture, it'll become a little bit clearer what CEP is doing. And in some sense, the worry about like, do we need to filter kind of our data first before we place it into the tree is kind of obviated by the fact that we're doing placement into a tree. And so hopefully this will be clear in the next section. But again, it really does depend. So we're not doing like a full phylogenetic reconstruction here. We're kind of taking the easy way out so that it's a bit faster. And even so, it still takes 20 minutes, right? So just wanted to throw that out there, that some of these questions will be made more clear in the next section.
Mike, did you have anything that you wanted to say about the reference databases? Or?
No, yeah, Evan hit it right on the head. Although I'll just quickly, one little question right here that I saw, which is about ITS sequences for the CEP. Well, we can talk about that later, but I'll just briefly say ITS is not amenable to phylogenetic reconstruction. And we can get into that after you watch the video and tomorrow, but I just wanted to point that out and plant the seed.
Great. Thank you. Let's see, just taking a look at this this chat. Okay, so there's a really great question here about how can we locate reference databases on the internet. So I would say the first stop here is the QIIME2 user docs. We have a couple of reference databases there, and then as well, pretty active discussion on the QIIME2 forum. Let's see. Here's another one here for you, Mike. So this is coming in here. Can we use QIIME2 to analyze trees that we've made in other programs?
Yes, you can. So if there's a particular tool that you prefer that may not be implemented in QIIME, or another method, you can, in the QIIME2 documentation, there's a page devoted to how to import various types of data. So if you already have a tree, I think most commonly a Newick formatted tree, you could import that tree. And the only caveat is to make sure that the labels on that tree, the tip labels of that tree, match whatever your corresponding sequence data is. So this way, when you're running Unifrac or other phylogenetic tools, that the tip labels on that tree match those what you have in your other data files for QIIME. But yep, so it's pretty agnostic in terms of being able to import the tree otherwise and use whatever tools that are there in QIIME.
Control room, I'm gonna share my screen really quick. So I just wanted to show you really quick to follow up on what Mike was just talking about here. So docs.qiime2.org. And if I scroll down here in the tutorial section, there is a tutorial called Importing Data. And if I click on this, it's a pretty long tutorial because it covers importing lots of very common starting point types of data into QIIME2. And so there's a little table of contents up at the top here, and you'll see right down near the bottom of this list is phylogenetic trees. And so if you had a Newick formatted phylogenetic tree, either rooted or unrooted, you could go ahead and import that into QIIME2 using a protocol like what is listed here. And if for some reason the importing tutorial didn't cover your needs, then QIIME2 forum is a great place to go for some more custom or tailored support. I'm going to turn off my screen share now. Let's see. So yeah, it looks like maybe folks are still running this command here, and I think we'll have plenty more questions after we get a chance to watch Mike's lecture about the phylogenetic reconstruction methods. So maybe we'll go ahead and break now. So I'll give you back those couple of minutes from the break that I took away from earlier this morning. So we will meet back up at 1 p.m. Mountain Time, and we will resume with this lecture on phylogenetic reconstruction. Also, we will probably have some more announcements right before we start that lecture. So make sure you come back in on time, and then we'll end the day with a general Q&A like what we did yesterday. So thank you very much. Welcome back, everyone. I hope you had a nice break. So hopefully by now your phylogenetic reconstruction with fragment insertion has finished. If it hasn't, please let us know in the philo tutorial topic in the discussion stream, especially if you've observed some kind of error or otherwise. And Yeah, I have a couple of other brief announcements here as well. So we are going to spend the rest of today on two things. We're going to cover the phylogenetic reconstruction lecture, and then at the end of that, we'll transition into general question and answer. There have been a couple of folks that have reached out to see if they could get some assistance with catching up. And so I think what we're going to do is put together a small breakout session during the general Q&A where 2 or 3 instructors will be available to kind of help work one-on-one with folks to get them caught up through the end of day 2. Now, if you do that, you won't be able to attend the general Q&A, but we will be recording the general Q&A, so you'll be able to go back and rewatch that either later today or tomorrow before the session starts. So please, keep an eye out for an announcement about that before we start the general Q&A, especially if you're a little bit behind and need an extra set of eyes to help get caught up. So with that, I'm going to turn things over to Mike. He is going to walk us through phylogenetic reconstruction. Thank you.
Hello, everyone. Welcome to the Microbiome Bioinformatics with QIIME 2 online workshop. Today, we're going to discuss phylogenetic reconstruction. My name is Mike Robeson. I'm an assistant professor at the University of Arkansas for Medical Sciences in the Department of Bioinformatics, where I currently focus on the human microbiome. So grab a cup of coffee or your favorite beverage, and let's talk about trees. Many of you are aware of the famous drawing by Charles Darwin called I Think, where he came up with the concept of a branching structure of species over time, or the variety of life as it goes extinct or diverges in creating new species over time. Hence the name of his book, Origin of Species. He was interested in how new taxa arrive over time. Now, what do we call this field of research? Well, phylogenetics. And if you break down the word phylogenetics, we're talking about the phylon or phyla, or in a sense, groups of things, whether they be tribes, clans, organisms, anything. So it's a collection of things, particularly in a population, and geneticos, which is the origin, a source of those groups of organisms. So we're interested in the history of these groups over time. One of the foundational insights we've gotten in the field of microbial phylogenetics is this idea of the 3 domains of life, discovered by Carl Woese and Fox. And what you see on the left is the phylogenetic structure or representation of the bacteria, archaea, and eukarya. And on the right, you see Carl Woese, and he's over a light table where he's looking at these old gels with blotches. And we'll discuss a little bit of history of these gels and how he was able to come up with this 3rd domain, or what he did to investigate and derive that there might be a 3rd domain of life. Now, in order to do this, in order to have characters that are meaningful in the phylogenetic sense, they have to be consistent. They have to be comparable in such a way that you can generate a sort of distance. The more different things are to one another, the more you should be able to quantify that difference. And he centered on the ribosomal or RNA molecule because he knew it was present in all microbes of interest that he was looking at. And he was able to look at differences of that molecule, the sequence of the molecule itself, and determine how similar or different these organisms were to one another. Well, how did he do this? Well, here's the only table or figure in the Woese and Fox 1977 paper in PNAS. And what this table is, it's a symmetrical matrix showing the similarity of representative eukaryota, bacteria, and archaea, which back then were known as archaebacteria. And you can see right away, right, as it makes sense, eukaryotes are more similar to each other than they are to E. coli or other bacteria, and even more similar to each other than they are archaea. Now, this table is what he kind of used to derive this third domain, or potential third domain at the time. And you can see right away, you can infer that, interesting, the archaea seem to be a bit more similar to eukarya than they are to bacteria. And this is kind of the first table showing from a sequence point of view, a molecular point of view, the similarity of these domains to each other. Now, how did he do this? How did he generate that table? Well, there's a variety of ways to do this, but often researchers used two-dimensional gels, and depending on the migration rate in these two directions, they were able to piece together bits of sequence of the rRNA molecule itself. Note, they're extracting the ribosomal rRNA directly. This is not DNA they're sequencing, but the RNA, as evidenced by the uracil notation. So these are the nucleotides that they're using as a kind of a GPS marker to put the other blobs in reference to. So what do I mean by that? So through a variety of methods in the lab and restriction digests and whatnot, you can piece together the sequence data along these two dimensions. And initially you have a dot with a G, a uracil and guanosine, and uracil and guanine. And depending on how far they migrate directionally from that point, you can piece together the sequence. So let's say I have a dot right here. Well, that sequence, that blob, is a GC. If I have a dot over here, then it's GAAC, right? Again, there's multiple ways to kind of generate these plots and piece things together, but over here you can see an example where you're basically drawing circles. You know, you have an overlay on your gel and you write on it and you say, oh, this blob is AGAU, this blob is GAAU, AGAC, right? And you could annotate what fragments. So you could kind of think of this as an archaic form of shotgun sequencing a single rRNA molecule. Now, couldn't really assemble the sequence. All you can do is figure out what characters or groups of letters together. In a sense, you could kind of view these as you would shotgun metagenome data, right? You could kind of think of these as k-mers in a sense, which is in a sense what they did here. So you have a set of characters of a certain length, and you just mark down for each one of those gels, and you have one of those gels per organism, and you mark down whether that sequence was in that organism or not. You put a 1 or a 0. And this is basically a simple character matrix. And there's a lot of tools where you can take a character matrix and you derive a tree like this. And then you can infer the relationships of these organisms given the character states, or in this case, sequence states that you have. So you could kind of view this, I always like to view this as a laboratory way of generating k-mer sequences. So what is old is now new again. Now, I've just skipped several decades of sequencing technology improvements, but here's kind of where we are now, using more modern and multi-gene phylogeny and comparative genomics approaches, where here's all of our eukaryotes, our various archaea, and then this big blob right here are the bacteria and the new bacterial clade, the candidate phylloradiation. Now you may be thinking to yourself, well, okay, fine, I know who's related to who, but how does it actually help me? Well, there's a lot of things you can do. So let's say you do an amplicon survey or a shotgun metagenome survey, or maybe you have other data that you can construct a phylogeny of. Well, you still may not have enough information about the molecule, or sorry, the organism, to know what it really is, but based on the phylogenetic proximity of that novel organism to things that you might be able to infer things about it. Maybe it's a pathogen and you want to know, well, how closely related is this bacteria to other pathogens, and can we infer what it might be susceptible to in terms of antibiotics? Do you know how it replicates? What metabolic output does it have? Can we say anything about the niche it lives in? Is it free living or symbiotic? So we could kind of start gaining insight into what a microbe is in this case, just by knowing where it falls in the tree of life. And this is just one of many things in our toolkit that we can use to better inform culturing recalcitrant microbes, right? So the Great Plate anomaly, we don't know how to grow and culture many microbes, and phylogenetics is used at least as one of those tools to help us figure out what can we do to make those organisms grow, given where that organism falls in proximity to others, right? So we might get some insight that, hey, this microbe likes to grow in this environment because, or we think that's the case because it's a sister taxa to this other microbe we already have information about. Okay, hopefully that was a nice little history lesson on the history of phylogenetics and sequencing and how it's evolved over time into the current state. Now, in QIIME, we offer a couple of ways of building and constructing phylogenies. One of them is a de novo approach, meaning we just take the data itself, we generate alignment, we do things to that alignment, and we build the tree from that alignment. Another approach is you can take a well-curated reference tree and alignment and insert your sequences into that tree. And this is very helpful when you are generating short sequences, as is commonly done with amplicon sequencing, and you want to try to extract as much phylogenetic information out of that as possible. And you can build the tree that way. We'll go through and compare both of these approaches, and hopefully you'll have better insight into what is the best way to analyze your data. Now, here's a listing of the common commands you might use for this section of the workshop. You have the alignment plugin, where you can make a de novo alignment with MAFT. You can mask that alignment, after which you can build the tree. You can use one of three algorithms or groups of algorithms: FastTree, RAxML, IQTree. And within RAxML IQTree, there's a bootstrapped version, and we'll discuss bootstrapping in a bit. And then when you're done making that tree, you often have to root that tree. Now, as a convenience, we also provide a pipeline which will carry out the entire process of making the alignment, masking the alignment, building the tree, and then rooting it for you, and capturing the outputs at each step along the way. And then we'll end on talking about fragment insertion. We'll do that because some of the problems that you'll see inherent in doing de novo phylogenies are sometimes circumvented by doing this approach. Now, depending on the data you have, you'll have to make the decision of which is a better approach, and we'll discuss those. De novo phylogenies. Now, here's a high-level view. So you import your sequence data, and you've learned, hopefully, about data types by this point. So you have unaligned sequences, which are grouped as the type feature data sequence. You can do a variety of intervening steps, and then you output a tree. Now, we have this tree, and we have the the tips of the tree, so where your taxa are, or your features, your exact sequence variants, your OTUs, whatever they are, they're the tips of the tree. And you have branch lengths, which often are indicative of substitution rates, how many substitutions in the DNA sequence occur. And if you want to know the distance between feature 2 and feature 3, you sum the branch length, this one here and this one here, and if you add these two, you get 1.1. That is the sequence distance between these two features. Now, the vertical line has no meaning other than to show the grouping. So there is no distance associated with the vertical line, only the horizontal lines. Now, these branch lengths, we'll get into how they're generated, but it depends on how you're calculating a distance, and there's many ways to do that. You have a node, which we're representing here, but this is also a node. This is also a node. And then you have the root of the tree, and we'll discuss the importance of roots right now. Okay. Now, many phylogenies, especially likelihood programs, or maximum likelihood programs like FastTree, IQTree, RexML, often output what's called an unrooted tree. And an unrooted tree often looks like this. And it doesn't provide a sense of directionality, especially since this is what we're often interested in. And that is, how do character states of the things we're looking at change over time? So when it's unrooted, you kind of don't have an inferred directionality of the relationships. Is this the oldest group? Is that the oldest group? Or is it this one? I don't know which way to go here. So you have no implied hierarchy. Now, if you pick a root, in this case, Nori was chosen as the root. Now we have a directionality. We know that this is the root of the tree, and then this is likely the out group, or the maybe often cases this is used as the most, say, ancient or oldest taxa, or relatively older taxa compared to everything else in the tree. So now you can start mapping features onto the tree and track changes over time, and as well as the clustering or the imposed hierarchy of these things. Now, this is just kind of a fun way of clustering things. You could probably come up with many other ways in which you'd rather cluster these. Maybe you can change the tree to reflect what your favorite and least favorite items are on this tree and make a tree that way. Okay. Now, before we get into making a tree, there's some considerations that we should discuss, and that's about homology. Now, homologous sequences share a common origin. Now, from there, we have to limit ourselves to the type of common origin they have. So an ortholog are homologous sequences, right? They share an origin, but they have a similar function. Now, likely what this means is that the gene in one organism is the same gene as the other organism. And the only reason why they're different is because those genes exist in different species. Now, you could have a case where you have paralogs, where the genes are different, not necessarily because they're in different taxa, but maybe because there was a gene duplication event. So that's what's shown here. You have a gene, that gene gets duplicated to X and X prime, but now the constraints of selection and drift might differ on this copy compared to this copy, and they will slowly diverge. And these genes may also co-diverge across the species, which is shown here. So there's a gene duplication event, and in this case, regardless of which of the two genes I use, I can reconstruct the species tree. In this case, both of these sections of the tree, the purple and the green, are showing that humans and chimps are more similar to each other than either is to a gorilla. However, there might be a case where either we don't have that information, or the gene was lost in that lineage, indicated by the dotted lines. So there are cases where we don't have the ability to make a proper tree, whether it be through technical reasons, i.e. we just don't have the data, or the genes were lost. So if I was to make a tree by not knowing that this was a duplicated gene and went under different evolutionary history than this gene, well, if I build this tree, then this tree is showing that humans and gorillas are more related to chimps, right? So this is a problem a lot of times when building phylogenies, or initially it was the problem with building phylogenies, is this: we had to deal with this issue of differentiating orthologs versus paralogs, and you always want to have orthologs. And that brings me to this idea of discordance. So what I just showed you about making that incorrect tree, because I might have a paralog, that's one way you could have an incorrect tree. The other is not necessarily that the tree is incorrect, but keep in mind that whenever you're making a tree from amplicon sequences, or any sequence, that there can be a discordance, meaning it's just a gene tree. We hope the gene tree follows the actual history of the species itself. So don't be too surprised if you're making a 16S tree or a cytochrome oxidase tree, gene tree, or any other gene tree, that there might be some differences in the topology that you find from the gene sequence versus what is known about the species. And this is kind of given by this example here, where you have 3 genes, a yellow, a red, and a blue. And each of these genes has a different history within the species. Again, gene duplication, gene loss. So just as you could have species go extinct, you could have genes within a lineage go extinct or diversify, duplicate. And here you can see there, among these 3 genes, there's 3 possible topologies, but only one of them, the blue one, matches the species tree. So always keep that in mind when you're building trees, is all you're ever really looking at is the structure of the gene itself. And you hope, generally you pick the gene because it matches the species evolution more directly than others. Okay. Now we're going to talk about de novo alignments. So before we even get to the tree, we have to overcome the issue of making sure we're comparing things correctly. So just as I discussed homologs and paralogs of the whole gene itself, well, we actually have to make sure we're comparing positional homology of the orthologous sequences, meaning just as we have that issue of orthology and paralogy of the whole gene itself, we have to have that discussion with columns of the alignment. How do we know that this alignment is likely the true alignment? So what we're saying here is that the T at this position in this gene is actually the same character T for this position in this other sequence. Here we have a mismatch. Why didn't we add a gap here? Well, we're basically stating that either one of 2 things, since we don't have a 3rd or 4th or 5th sequence to give us a direction, just like rooting a tree. So just from these 2 sequences alone, we don't know if the A became a G, or did the G become an A. Either way, we don't know the order of the substitution that occurred. And there's different rules and criteria and many different ways to kind of derive a sequence alignment. Here is a very traditional approach called dynamic programming. This is kind of the method that BLAST and many other tools use, and this is just showing a simple case for 2 sequences. And there's a lot of parameters and rules that you could apply to this model to dictate, well, what is the criteria I need to insert a gap? What is the criteria I need to extend the gaps? And what is the criteria to allow a mismatch? In other words, this is the crux of the issue. How do you decide that this should be aligned in the same column as a mismatch, or whether or not you consider it different enough to insert a gap? And this is very big problem, especially with dealing with very ancient sequences that varied a lot. How do you infer these? Is basically the biggest problem with sequence alignments. But just know there's many different programs. MAFT is the one that we'll use here, but there's a lot of different tools with different assumptions about the data and parameters you can change given the type of sequence data you're using. Here, today we're mostly talking about DNA sequences, but there's different criteria for amino acid sequences that can be used as well. But the idea is, how do we do indels and mismatches? Oh yeah, sorry. Indels is an insertion and deletion. So what we're discussing here, again, we don't have directionality with these two sequences, but this sequence, you could say, has a deletion. In other words, it lost a G and a C, so there's no bases there. Or you can say this sequence has an insertion. It gained a G and C. Now, without older or other sequences in this alignment, we don't know which way this occurred, so we just call it an indel. That set of columns in this alignment is just an indel column. Now, there are many ways to reduce ambiguity. I won't spend too much time on this, but you could have guide alignments where you have maybe you have a nice curated set of sequence alignments that you want to just simply map your reads to because you trust that alignment. De novo alignments can sometimes have problems. You might get a slightly different alignment every time you add a new sequence. But if you want to minimize that effect, you could have a reference alignment that you're mapping sequences to. This example here with rRNA data or other genes where you actually have some sort of secondary structure, you could use that information to guide the alignment. And it actually helps you determine whether, or the model determine, whether it should allow mismatches, gaps, or mismatches or indels. So as you can see here, you have sequences here. This side of the molecule has complements to the other side. You could have, in a sense, insert gaps into the alignment because these are loops. So depending on the stem and loop structure and the information you get from that, you could add that to your model to make a better de novo alignment. Well, how is this done? Well, there's many ways. Here's just two. We won't spend too long here, but the idea is you've heard of NAST, nearest alignment space termination, and then SINA, which is the Silva incremental lineup. But the idea is you can take your query sequence and you can find either a set of sequences that match that query and then use that closest match to help guide the alignment. Great, you have a template that you can map that to. Or you can create a model, and there's many ways to do this. This is the Silva approach. There's others like hidden Markov models that can be used, but you generate a sort of graph or a model of the alignment, and when you insert your new sequence, it tries to follow the set of rules. For example, we have A, G, and then we can either go to G or skip the G and become an A, and that skip G to A basically means there's a gap. Here we can go directly from G to T, or G, CG, an insertion here. Now, again, there's many ways to do this, but this is a way to help guide, especially more problematic sequences that are more difficult to align, and if you have a way to curate the sequences, you can do that as well. Now, after you do an alignment, whether you use a reference-based approach or you're using a completely de novo approach like using MAFT, there might be issues of the alignment algorithm, or maybe the sequence is just too noisy and you can't construct a great alignment. And there's probably concerns about, all these columns really truly homologous positions in the alignment. Are there really things we should be comparing? Now, the approach of applying a mask, often called a Lane mask, proposed by David Lane in 1991, is to help remove these errors where you kind of might introduce more error in making the tree. So you could probably make a better tree by removing these noisy regions of the alignment, simply because you don't really know if those bases in that column should be compared to one another. So you remove them. Also, it's used to remove these phylogenetically uninformative sites. Again, there's many ways of masking. Sometimes you can mask things that are just exactly the same or not. And this depends on model substitution models and whatnot, which we'll get into a little bit in a few minutes. Often, as with amplicon sequencing as well as genome sequencing, if you have repetitive regions or homopolymeric regions, you might have, say, a session with 10 A's and another sequence with 8 A's. Well, where do you put the gap? They're all the same. And that can matter because other sequences may not have all those A's in that region. So it becomes difficult to know where to introduce the gap or allow a mismatch in that sequence. Or you can have a repetitive sequence. Maybe I have here I have 4 TA repeats, but another sequence I might have 8 TA repeats. So where do I put those gaps? It's hard to know. So we just remove them. And you can kind of, removing these helps clean up the phylogeny or hopefully make a better phylogeny. Now, keep in mind, depending on the alignment approach you use, might alter the structure of the alignment and may change how that alignment ends up being masked. I could use one tool to do an alignment, and it might give me a different alignment than another tool. And then when I go to mask them, I might be masking out different things. So be cognizant of that. Now, there are caveats to masking. The most common arguments against masking are if your mask is too aggressive, you might be removing too much information. And very often you make the sequences more similar to one another, or in some cases, even identical, meaning that you're losing some phylogenetic resolution. Now, this may not be important in all cases. If you're only comparing, say, at the community level, this may not be too much of a problem. But if you really care about the actual phylogeny of what you're looking at, then you might be detracting from making use of exact sequence variants because you might have 2 sequence variants that will become identical after you mask. Now again, that's only important if you're really wanting to look at the actual phylogeny of your exact sequence variants. If you don't really care too much about that and you're just generating a tree for community level comparisons, say UniFrac or FAES phylodiversity or any other phylogeny for metric for the community level, you might be okay. But just keep this in mind when you're deciding on how to mask or how aggressive you should be masking. So ultimately, it really depends on the question you're after or the question you're hoping to answer. And you should balance out the level of masking or not mask at all. And you can do some iterative runs. You can try making a tree without a mask, or you can make it with different levels of masking. So there is no one size fits all. Depends on the marker gene that you're using and the question you're after. Okay, now that we sort of kind of got our alignments worked out, the next step is to build a tree. Now that we have our alignment and we have hopefully our positions lined up appropriately, you have to now build the tree. Now, with some of the phylogeny programs included in QIIME, they only have 1 or 2 models that you can use. One of the programs that I like to use often is IQTree, and it's got a couple hundred models that you can choose from, and it will look at your alignment if you want, assess what the best substitution model is for that given data set, and then build a tree given that model. Now, what is a substitution model? Well, it's about how bases can interchange or substitute from one another. The most simplest model, say the Jukes-Kanner 1969 model, shows that any base can equally change to any other base, regardless if it's a transition or a transversion. So what is a transition or transversion? Well, if you remember your basic biology memory tool here, pyrimidine, cytosine, uracil, thiamine, or picut, and purine, adenine, guanine, pure as gold. So if you're changing between any of these bases or any of these bases, it's a transition. But if you're changing between bases, transversion. So here you have another model where you have T and C's equally changing towards each other, or A and G towards each other, but less often so between. Here you have a different case where the model is slightly different, where you have T to C, C to T. Here you have A more often going to T than the other way around. Now there's a few hundred models that you can potentially choose from, or models that your data may fit. But the idea is, once you have an appropriate model, or you have one based on some prior knowledge, you then can do bootstrapping. Well, what is bootstrapping? Well, the name comes from holding yourself up by yourself. You grab the bootstraps on your shoes and you hold yourself and you float in the air. So basically what you're doing is you're sampling with replacement, meaning you're sampling columns of the alignment with replacement, which means I can sample the same column more than one time, and I create a pseudo data set. And the pseudo data set here is the same length as the original alignment. And from the pseudo data set, I build a tree. And I can do this, when I do this, I often do this 1,000 times. So I have 1,000 bootstrap data sets. And for each of those, you build a tree for each data set, which is why bootstrapping can often take a while. You're inferring hundreds or thousands of trees. And then at the end, you make a consensus tree from this output. And what that means is you are going to come up with a way to quantify how well supported the branching pattern or branching structure, or sometimes the split structure of the tree. In this case, I assume the 67% is falling here on this branch. So what we're saying here is after this branch or this split, so you could think of splitting this part of the tree from this part of the tree, seek A and seek B appear together among all these permutations 67% of the time, right? Meaning the other, 23 or so percent of the time, you might have seek A and seek C, or seek B and seek C, or anything like that. All that percentage means is these two taxa appear after this split 67% of my pseudo replicates. So no matter where, so when you're looking at these percentages, bootstrap percentages specifically, there are other measures of support for nodes, and they mean different things, but specifically for bootstrapping, whenever you see a bootstrap number, it's basically saying any and all taxa that appear after that split appear there that percentage of time. Normally a good support, and it can vary depending on who you talk to. Often with bootstrapping, usually 70 to 80% is a pretty good support for that node, or that branch, I should say, that split. Usually if it's less than that, you can say, I'm going to collapse this. You get rid of that. And then you can say, I can't resolve the differences between seek A, B, and C. Okay. So now you know what kind of happens behind the scenes when you're bootstrapping a tree and you're getting confidence of the nodes of that tree. Now, again, that was all the de novo process. Now, here's an approach called fragment insertion. Here's the paper for it, and if you want to read more about it or the tutorial, you can click on the GitHub page for Q2 fragment insertion. So what does this do? Well, let's kind of refer back to our de novo approach. So normally, right, we might have a reference database with full-length 16S sequences. But we're only, in this case, we're going to use the V4 amplicon region. Now, you can see right away that the amount of phylogenetic information we have is drastically much less than the entire full length of the gene itself. And that can cause some problems. One is here the red lines are our amplicon sequences, but only 4 of which have exact or nearly so representatives or sequences, full-length sequences that it matched to in the reference database. The rest do not have a match or a close match to those in the reference database. And here is the crux of the problem. What do we do with these sequences that don't have a direct match in the reference database? Well, there's 3 things you can do. You can do a read recruitment where, depending on some dissimilarity criteria that you allow, it could be 100%, 99%, 97% similarity, and you only keep those reads that can recruit to the reference data with that sequence similarity threshold you set. However, you end up discarding a lot of real data. So these aren't necessarily bad sequences or chimeras or anything like that. They could be real, but you're discarding them simply because they don't match well enough to the criteria you set. The other approach, the other end of the spectrum, is you can keep everything. However, just like we discussed about a gene tree not necessarily matching the species tree, a subsequence or subgene, I guess we can call it a subgene, but a subsequence of a larger sequence can also not give us the same tree. A full tree given by the full-length sequences is going to give us a different tree than that just based on the amplicon region. Why is that? Well, I could have a case where I have 2 full-length sequences that are extremely different from each other over the full length, but for whatever reason, they're nearly identical over this region. So in the de novo tree, it might make those sequences appear more similar to one another than they should be. I could have the opposite case where my full-length sequence is almost identical, except for maybe that bit of the V4 region. So now I could be inflating differences. I could have that sequence here and another sequence here. So the issue with doing de novo short reads is you could either overinflate differences or underinflate differences between, because really the tree is just reflecting the information content of that small amount of sequence. So what can we do to get around? Neither of these are really ideal when dealing with short reads. So how about we combine the two and do something called an insertion tree, which is combining both of these. So what are we doing? Well, first of all, the best case is we insert something that matches fine. We don't have anything to work out there. The issue is, what do we do with these reads that don't have an exact match? Well, what you can do is you could figure out through an iterative procedure where they fall on the existing tree and insert them appropriately. Well, how is this done? Well, it's done by an approach. This is what's going on with the fragment insertion approach. It's taking a technique from de novo alignments called SATE, which is the simultaneous alignment and tree estimation. Now, one thing I didn't tell you before is for very tricky sequence alignments, it's very difficult to know how we should align them before making the tree. So we can really only know the true sequence alignment unless we have the phylogeny. But the problem is we can't get the phylogeny without the alignment. So it's almost can be a case of circular reasoning, but not quite. And again, this is only a problem for ancient sequences that are very difficult to align. And there are many other tools out there that do this co-estimation of the alignment and the tree. And it's this iterative process where we have an initial alignment. Some programs will come up with an equally likely set of alignments to come up with an equally likely set of trees, and then use that to infer a new set of alignments, and so on and so forth. Again, there's many different ways to do this approach, but the idea is you're kind of trying to leverage both bits of information, or at least as much as you can extract from that. And that's what this simultaneous alignment and tree estimation is doing. Now, The part we're going to discuss more is SEP, which is you're taking this iterative approach to help you better place fragments into a tree. And in a general sense, what we're doing is we're taking subsets of the tree and the corresponding subsets of the alignment to kind of work out where the sequence should go. So let's say, if you look at the tree on the right, here's our base tree. We have a short read, and on our first round through this loop, it inserts the fragment here. Now it's basing that off of a local alignment, or its initial round of making a local alignment and only making a subtree. So it's aligning only to part of the tree, and it's placing it here. And it goes through an iterative process, and I'm hyper exaggerating the process here just to make the point, but you can see that, well, through the next process, it might decide to insert over here. And then it goes through a process again, and then it inserts it here. And let's say it goes through a few more rounds of iteration and it doesn't change. This is likely the place it belongs, and now that is where your fragment is inserted. Now it could easily go the other direction. It could start off here and maybe end up coming off in the more base part of the tree. But that is, in a nutshell, what it's doing. It's iteratively trying to refine where these fragments go. Now, this is helpful. Now, as you can see here, here's a PCOA plot of your data. And when you do, say, a de novo approach of mapped and fast tree, you can get some artifacts where samples don't necessarily show up where they should. They're showing up together, probably because they're so different, or, as I said, given the amount of information that short read, right, we could hyperinflate their similarities or their differences. In this case, I'm assuming we're hyperinflating their similarities, which is why they're all showing up here, as opposed to the rest of where they should be going, as indicated by the color. And these two other plots are two different test cases where they're using the fragment insertion approach, and you can see, you can kind of recover where these points should fall in your plot. Now we've completed our approach here. Oops, I put my my highlighter here. It's supposed to be around data resources. But if you go to the QIIME page and you want to make use of the fragment insertion, there's two trees you can make use of. There's one based on Silva 128, another based on Silva 13.8, and you could insert your trees there. There's other information on the data resources page as well. Check out these two tutorials. The current version we're on now is 2020.6, but if we're using a later version, just change that to the current version. And the Q2 fragment insertion tutorial here. And again, if you want to know more information about these tools, just type in the plugin and -help. And hopefully you've learned a little bit in the general sense of how to do and how to build trees and how to construct phylogenies and all of the caveats you should think about when doing so. So that's it. It was my pleasure to discuss this with you, and I hope all your trees are fully resolved or nearly so. Take care.
Welcome back. Thanks so much, Mike, for that lecture. And I've got Mike standing by here to answer any questions that you might have. If you do have questions and you haven't asked them yet, head on over to the questions broadcasts, or the discussion stream, and send your question in the Philo lecture topic. So I guess I wanted to kind of circle back. Mike, was there any questions that had come up after the lecture, or after the tutorial earlier that maybe now we should revisit? Now that we've had a chance to go into some theory and background?
Sure. So one of the questions, and I believe I put a response to in the discussion, was a question about ITS sequences. So just to revisit that topic again, there's a lot of sequence data out there, or marker genes, if you will, that aren't very amenable to phylogenetic reconstruction, either because They are hyper variable. In other words, they have a very high substitution rate. So what you learn when you study phylogenetics, there's markers that are really good for population level, and there's markers that are good for deep phylogeny. So there's a lot of genes that are very conserved, and all you can do is maybe resolve the family level. Then you have other genes that are like population level, and you can only resolve within population differences or near highly related species differences, and beyond which everything looks like a comb, right? All the taxa, you can't resolve the network structure later back in the past, further back in the past. So ITS is one of these where you might be able to do a tree and a phylogeny if you're, say, looking within a genus, right? But you can't globally align it across all taxa because there's so many substitutions and changes that you could have convergent substitutions. In other words, you have totally different taxa mutating or substituting a base that have nothing to do with their linked ancestry, but just due to the odds they switch to the same base. So to get around this issue, there's a plugin called Ghost Tree, which you can take your ITS sequences and you can map them onto phylogeny. I believe it's an 18S gene phylogeny, where you have a database of 18S gene sequences, and it maps to known sequence from the same organism for the ITS marker gene. And what you can do is you just map those ITS reads to that data, and it'll then map that data to the 18S, which you have the tree for, and you can get around it that way. Can't remember if there's another question in there somewhere, but that'll hopefully answers that question.
Thank you. Let's see. I'm just going through all of my various streams of data here to see. There was one question here that I think got put onto the end of day queue, but maybe we can ask it now since it's tied in. So it's about the semantic types and data formats that we should be using when we import a reference database into QIIME 2. And I can say some words on that if you'd like, or you.
Yeah, I guess it depends on what we're going to be doing for what is going to be used for the reference database. So generally, when you're importing a lot of these data sets for references, you kind of, in a way, mostly import them like you would your normal sequence data. So it's either feature data sequence for your reference sequences, or if they're aligned, feature data aligned sequence, same thing with taxonomy. Just import the taxonomy. So generally they're the same that way. So in a few other cases, like if you were to make your own SEP reference database, that's a different set of semantic types there. I guess I'll leave Matt to maybe clarify some of that.
That's thank you. I think that's more or less what I was going to say. Is it sort of depends on the direction that you're going. You know, if you're going to be using fragment insertion, SEP versus some other direction, like one of the methods wrapped in Q2 phylogeny. But it's definitely the kind of thing that we'd be happy to help with on the QIIME 2 forum if you need some specific guidance. I'll also just mention briefly with respect to SEP and fragment insertion, the databases that we currently have published for those up on the QIIME 2 docs are sort of vetted or approved from the team that created SEP. And so there's some discussion about doing benchmarking and whatnot for other databases. That's not something that all of the QIIME 2 developers necessarily have insight into, but there is some pretty active discussion into improving the tooling and whatnot. But it is something that I've heard them, in particular, Stefan Janssen, mention on the QIIME 2 forum about some concerns or caveats. You maybe don't want to just jump into using Q2 fragment insertion with a home rolled database without spending some time to sort of verify and validate. Let's see. So I've got another question here. When choosing primers for a marker gene, how do you balance the phylogenetic information content against the richness of existing databases?
Okay, so that's a tough one, and a lot of work goes into determining that. And ultimately, at least in my experience, this is where, like anything else, right? It depends. Like, what is the question you're after? Are you interested in just a specific group of bacteria or fungi or animals, for that matter? What is it you're trying to differentiate? So this is I'll try and find some papers to put up here, or maybe some of the other instructors already know them and they can post them. But there's a lot of discussion. For example, if I'm doing the skin microbiome, well, it appears, if I'm remembering correctly, the V1, V3 primer set does very well for the skin microbiome because it can best differentiate among the taxa found on the skin. For gut microbiome, you might want to use V4 or V3, V4, right? So having some knowledge of the type of organisms you're going to encounter in your data set can help you decide what region best differentiates among those particular taxa. So that's the approach I use. So it does take some ground truthing and looking that up, but that's generally how I would go about doing that.
Thank you. Let's see. Doesn't look like there's any other questions there. Maybe this would be a good time to kind of transition into the next segment where we move into the general Q&A, and then if people have additional phylogenetic reconstruction questions, we can certainly add those into the queue. So thank you, Mike. And let's see, I have just a few quick announcements before we move into the general Q&A. First off, the fragment insertion step that we ran before our last break, I think everyone that we've heard from has finished that or is close to finishing that. If you are still working on that, please let us know. We can help you get situated before moving on to the next steps tomorrow when we resume the tutorial. And then the other announcement here is during this general Q&A that we're going to conduct for the next 60 minutes or so, we're going to host a parallel breakout session. This is a little bit impromptu, but a few of the instructors are going to set up a little chat room in the discussion somewhere to get folks together who are not caught up yet on the tutorial. So if you're stuck on the steps that we covered either earlier today or yesterday and need a little bit of extra assistance getting through that, you'll want to go over to discussion. Let me see, have they posted anything about this? Can I get one of the instructors to just post an @everyone announcement to let folks know which room you'll be hosting that in or which section. Now, obviously, if you're attending that session, you won't be able to watch the Q&A live, but we will be recording this, and you'll be able to play back this Q&A later today or tomorrow before you resume the course. So it looks like Chloe just posted a message under discussion. So she created a new topic called catch up. So if you are behind and would like to get caught up through the end of today's tutorial material, please reach out to the instructors there and they will work with you to try and get you situated. So with that, we're going to move into the general question and answer section. And I want to invite the instructors that might be waiting in the wings into our studio room. Actually, maybe if I can the control room send out an instructor notification over in the studio chat? Thank you. And then I am going to pull up my question queue. So there are a few questions in the queue that we had set aside for the COPE lab. And so when I see them jump into the studio chat here, we'll start sending those questions their way. So let's see. It's like a wall of text here. Let me see what I can find in here. Okay, so during the demultiplexing tutorial earlier today, we had a question come in about the Fred offset. So the question is, if you specify the wrong Fred version when you're importing your sequences into QIIME 2, will you get an error about it? Assuming here an error from a QIIME 2 command. I'm wondering if maybe Bod, would you like to address that, or?
Sure. Yeah. So good question. I think I may have written an answer in there, but just for continuity here, I'll go again. The likelihood is that you will see an error because there are certain characters that are expected in one format that are not present in the other. However, that's not a guarantee because there are crossovers. There's an overlap between the available ASCII characters in both types. So if for some reason your sequences are all within the quality scores are within that overlap, then it won't know the difference. It won't be able to sort of differentiate between the two formats, and it will raise an error, or it wouldn't raise an error, and you will sort of slip through the cracks there. So yeah, the probability of that happening is low, but it is still possible. So we definitely recommend looking at the data, talking to your sequencing facility, and being sure of what format you have before going through that step. There are tools out there that try to predict this, but they run into the same shortcomings that if you have these rare instances where your sequencing, your quality characters are within that overlap, then it'll slip through.
Can I add one thing to that?
Please.
Yeah, one of the issues with this, and maybe folks already talked about this today, but there's just no way to tell that information is not present in those FASTQ files. And so like, if it was, then we would always be able to put an error message up, but we wouldn't need the user to have that information if it was in that file in some way. So it's definitely something we, try and catch, but it's just not foolproof.
Yeah, thank you. Emily, did you wind up getting connected here? See you in the chat. Maybe not. Okay. Let's see. I'll move on to something else. Okay, so we had a question earlier today, I think during the denoising tutorial about filtering chloroplast and mitochondrial sequences. Greg, is that something you could say a couple of words about?
Yep, definitely. Okay, so yeah, so the as you probably know, the 16S rRNA PCR primers, will often amplify chloroplast and mitochondrial 16S. And so, depending on what sample type you have, they could end up being a large fraction of your sample. So, for example, if you are sampling the surface of leaves, you might end up with a lot of chloroplast sequences in there. If you're sequencing like a low microbial biomass site in the human body, like lungs, for example, you might end up with a lot of mitochondrial sequences in there. The way that we typically filter those out in QIIME 2 is after the taxonomic assignment step. And so those sequences will typically get annotated either as chloroplast or mitochondrial sequences. They will both show up under the proteobacteria, if I understand correctly, or if I remember correctly. And we have an action in QIIME 2. It is QIIME tax. I believe it's QIIME taxa or QIIME taxa filter, maybe. I will point you at a tutorial right now in reply to this question that shows exactly how to do that. But there is a command that is specifically for filtering a feature table based on the taxonomy of features. And so you can filter both the mitochondria and the chloroplast sequences. Never a bad idea to do this. If it's, if they're making up only a very minor fraction of your sample, it's probably not very important. Like, I know I've probably forgotten to do that from time to time. And it probably doesn't have much of an impact on the results unless it's starting to, make up a considerable fraction of your sample. But it's never a bad idea. You could just make that part of your typical QIIME 2 workflow to filter mitochondria and chloroplast sequences. And I'll go reply to that question right now with a link to the tutorial that I'm thinking of.
Cool, thank you. Mike.
Yeah, I just wanted to add to what Greg just said, particularly with taxonomy. So sometimes the taxonomy, depending on the database you use, if you want to remove chloroplast or mitochondria, it's always obviously a good idea to remove those. Sometimes folks may forget, and when you're trying to do a general summary of taxonomy, say at the phylum level, and you're doing a summary plot of just cyanobacteria or proteobacteria. So the oddity of organelle taxonomy is such that the deeper phylogeny taxonomy string is representative of, say, the bacteria. But so if I just filtered a data set and said, well, I want to keep all of my cyanobacteria, well, guess what? Chloroplasts are within the clade cyanobacteria. And there have been some published papers out there that make some claims about cyanobacteria, but then when you look at the actual data, you find out they basically forgot to remove the chloroplast sequences. So be very careful about that. So I just wanted to point out that warning. So.
Great, thank you. So it looks like Emily was able to get into the chat here. Hello.
Boy, there we go. Am I muted?
We can hear you.
Great.
Welcome. Let's see. So I am looking at our questions end of day stream right now. And I think I'm going to probably jump around here a little bit. So we had a question come up yesterday that we kind of deferred to when you were going to be around here. So Greg had mentioned in his kind of introduction lecture about how sequencing is incredibly powerful technique for microbiome research, but culturing continues to be like pretty informative as well. And so the question was, what about when it's difficult to culture an organism or organisms? And I'm trying to remember, Greg, if you had any specific follow-ups or questions for Emily related to that. But we were basically we're just kind of thinking, well, let's get someone with some lab perspective in on this question here. So, do you have any thoughts on that?
Yeah, I didn't have anything specific aside from really like whether, Emily, you had any thoughts about how sequencing could be helpful for culturing. And if not, I know Mike also I think Mike had some thoughts on that. I saw some follow up in the chat. So would love to just hear what both of you think about that.
Yeah, I can jump in. So my first thought is that most bacteria in a lab setting are very difficult to culture, if at all. So, if we can sequence and recognize which bacteria are present in a sample and then move backward and create an environment for certain bacteria. So if they need a certain media or they need different conditions. So being able to recognize what taxa we're focusing on or what's present and what we can culture before we try to culture, if it's not already known, I think that a decent amount of our model organisms, we know that, strep is going to be highly present in the airways. And so in that case, I think that culturing can be efficient in the beginning, but I think if it's a newer organism or we just don't really know a lot about it, or a newer sample type, then it can definitely be helpful to understand what's present before going about culturing and getting the environment that we need in the lab and the media that we need.
Cool, thank you. Yeah, speaking of the airway there, we had a couple of questions come up about low biomass samples. So I think, one question was sort of just like, do you have any thoughts about sampling and techniques? And then there was also another follow-up question about like contamination specifically related to these low biomass samples.
Yeah, luckily our lab works with a lot of those. So I think that there's a couple ways to go about it. The first thing that you have to think about is how you're going to extract the DNA. And so if you're expecting it's going to be low biomass, if you're using a kit, it can be really helpful to elute less. So say we're just going to make up a number to make it easy. There's 100 nanograms of something. And if we can elute it into less microliters, then you're going to have a higher biomass, but less sample to work with. So I think that's kind of a caveat, is that you can elute less, so you'll end up with less sample in volume, but more in concentration. So that would be one of the first things I would suggest. There's also different kits or different techniques for extracting DNA. And so I know that in the past, our group has used the phenylchloroform extraction, which can yield higher biomass and higher quality, but it is more difficult, and it's not just a kit. You have to go out of your way to get the different components for that. So I think that would be something to consider as well if you have something that you're struggling to get enough DNA from. And then as far as contamination goes, that comes a lot more down to the PCR part of it and how many cycles you're running it on. So when we have lower biomass, we will sometimes run 35 cycles for PCR instead of 30. It does increase the chance of contamination if it's there, because you're going to amplify the contamination as well as your target DNA. So that's also something to consider. But playing around with the PCR cycles and the temperatures can sometimes be a benefit if you have a very low biomass sample.
Thank you. Let's see. So we also
looks like Mike had.
Can I add something?
Thank you.
That?
I'm getting an update here from the controller. Mike, jump on in.
Okay, sure. Yeah, just to extend what Emily said, I heard one approach that's using, and maybe the other instructors can comment, to avoid the over amplifying of a sample is some researchers will do replicate PCRs where they'll take a sample and they'll do like maybe 20 or 25 cycles or 30, and then they pool those together. I don't know if anybody else has had experience, but that's something else you might want to look into. I'm trying to remember the citation I have, but I figured I'd mention it. But there's a lot of, obviously there's a billion ways to deal with these issues. But anyway.
Cool, thank you. So Emily, we have another question here, and this came up, I believe, during your lab protocols lecture this morning. Someone asked, I'm pretty sure the extractions done on our lab have not been performed in the BSC. How concerned should we be about contamination?
This is something that we've discussed a lot within our lab group. We do BSC extractions to protect our samples more than us. I know that other groups do benchtop for microbiome samples. Our lab does a lot with many different types of bacteria, and so we're more concerned about floating amplicons in the lab and general contamination from other groups. And so we do it more as a precautionary thing. I think if you can do it, it's always better. However, I recognize that's not always an option, and so I don't think it's a necessity.
Emily, I've got a quick follow-up for you on that. So one of the things that's unique about, or not unique, but relatively unique maybe about our lab, is that it is shared between multiple different research groups. Does that impact like your decision to use the BSC, or just curious, like if it's because it's a shared space where people are working on pathogens and things like that as well?
Yeah, I think that definitely has a lot to do with it. Sometimes labs, you have a smaller lab space, but it's all your own projects. And so I think you can control contamination a lot better in that case. We also have a very busy lab, and there's constantly people working on bacteria. And so I agree. I think that a lot of that has to do with our lab space and the specific conditions that we work under. And having the ability to do it in the BSC just makes it a little bit safer for our samples.
Real quick, can I ask you to elaborate on the BSC acronym for those of us not in the know here? Thank you.
Yes, of course. So BSC stands for Biosafety Cabinet, and then there's different levels of, biosafety level 1 through 4, depending on the work that you're doing. So our BSCs are BSL 2, but we do BSL 1 work in it. So we're being more cautious than we would need to be.
Cool. Bod, did you have something you wanted to jump in with on this BSC and contamination chat?
Favorite chat. So yeah, of course, everything that was said, I just wanted to take it even one step further as someone who wasted a few weeks troubleshooting contamination. So not only is it really important, of course, to protect your own samples, amplicons, because they're DNA, they're really stable. They can linger for a very long time, and they will completely ruin other people's projects. So as much as you want to protect yourself, but also consider that you're going to really mess up other people's project if that contamination reaches their work. So with that being said, if you can access the BSC, I strongly recommend using either bleach or DNAs, whatever you have access, before and after your procedure. In my old lab, we used to have a dedicated room just for amplicons because they are such they're such a menace to deal with when there is contamination of it. We wanted to keep it away from everyone else's work as much as we can. So if those are not an option to you, strongly recommend decontaminating using bleach or DNAs to remove any amplicons that may have sort of gone out on aerosols or what have not. It really will other people will thank you if if you don't thank yourself.
Yeah, and just to clarify other uses that you can use the BSC for, we recently did PCRs for a very sensitive project in the BSC. So we were extracting, well, the other group extracted, but they extracted in BSC, and then we PCR'd in a BSC. And then when we do PCR in a PCR hood for our normal samples, I still UV it and ethanol and DNA away just to be extra precautious. So I think anything you can do in microbiome work to prevent any further contamination is worth the extra work and money.
No doubt. We had one or two questions come in throughout the day about like thoughts on specific kits. I'm not sure, if you want to comment on specific kits, or if you have any thoughts about general trends or whatnot, but I'd love to hear what you have to say.
Yeah, so I think this question came up in the last one, and I just want to clarify that I'm not endorsing these particular kits. These are just the ones that I've used and what I've used them for. So we have 3 different kits that I've used. One of them is the DNA Power Soil from QIIME, and this one only extracts DNA. And so if all you're planning on doing is 16S, it can be really effective and is a nice kit to use for that. For a lot of our samples, we like to do qPCR or other things that we might need RNA or DNA for. And so we'll use most of the time a kit that will elute both DNA and RNA. So we use the QIIGEN All Prep most of the time for that. And then finally, we have a Kingfisher machine now at our work facilities. And so that helps with high throughput projects. So for example, my project had 2,000 samples that we sequenced. And so being able to extract 96 samples in a couple of hours was really helpful. And so if you have access to that, there's, what is the name of it? I think it's called the MagMax pathogen kit, but it also does DNA and RNA. And so those are the ones that I've had experience with. I think depending on what you have access to and what your project requires, you would use one of those based on it. So those are the 3 I've used. They've worked well for all of our samples. So we've had, mostly colon and fecal, which are high biomass, but we've also used these on the sinus and the lungs and vaginal. And so those are lower biomass, and they've worked for all of those.
Awesome. Cool. Thank you. And thank you for the caveat about the endorsement or lack thereof. So I'm going to pose this, I guess, to you, Emily, first, and then see what other instructors want to jump in. But there's a question earlier today about triplicates or replicates of samples. I think this one was specifically about triplicates. And so the question here is, I couldn't tell if the intent is to sequence each replicate separately to check for repeatability or to pool replicates before sequencing. Yeah, curious what you think about that.
Yeah, so the way that we do it for PCR is we run in triplicates and then we end up pooling all of them before we move further. And so the idea is that we should be able to get 3 separate reactions. And I think a lot of it is a preventative. If one reaction fails, you're still having 2 that work, or just confirming that you have a reaction that works or doesn't work by doing it 3 times. So we pool it, and then we move on to the next step, which would be to quantify the PCR amplicons. So we do not pool the triplicates separately. We pool them together, if that makes sense.
Yeah, totally. So anyone else have anything they want to add or comment on about PCR replicates? No, I'm sorry. I'm looking at my chat here on the side. Nope. Okay. Let's see. I have so many notifications on my screen right now. Where am I? Okay, there is a question here for Emily. Can you comment on your experience on contamination related to sample storage methodology? Snap freezing versus preservative agents, storage time. And I suspect Greg might have some follow-up for that as well.
Yeah, so in my experience with my samples in particular, because those were the only ones that I collected and stored all by myself, we used the, what is it called? I have all of the different RNA things in my head, but there's an RNA storage buffer, and I will think of the name of it and throw it in the chat because I cannot remember at the moment.
Is that RNA later?
RNA later, yes. All in my head was RNA away, and I knew that was not right. Yeah, so we use RNA later, and this is to help preserve our RNA. So I'm not really sure if you're just looking to extract DNA. In the past, we've also just put certain samples in PBS, and we store it at the -80, and we haven't had any issues. Yeah, I can't think of anything else. Once it's amplicons, you can put it in the -20. But as far as storage of samples right away, we either store it in PBS or in the RNA later.
Cool, thank you. Yeah, Greg, I was reminded of, I remember we had a discussion once about, maybe it was the FMT project. We had all kinds of like mail-in samples and whatnot. Just, yeah, wondering if you have some thoughts.
Yeah, I mean, as far as storage for stool, we basically try and get it in the -20 as soon as possible. Ideal is just getting it in the -20 right away. There can be complications with that sometimes. Like we have done some projects where samples are shipped through the mail. And the longer they're in transit, the more different they start to look from samples that are frozen right away. -80 is not, in my experience, not necessary for fecal storage pre-processing.
Anyone else want to add into that?
Just commenting that that's probably more specific to DNA, but if you are collecting with the intention of doing RNA, the RNA can still degrade even in -20, and of course even in -80 at a much slower rate. But it really should be sort of decided. The important thing is that once you decide on a protocol that works for you, because your stool maybe is being shipped from across the country, or maybe you have participants right in the sort of the lab providing samples, hopefully not in the lab, but somewhere else privately. But the idea being that once you have your protocol chosen or your kit selected, make sure you stick with that protocol and kit throughout the entire project. Because even those slight differences, if some are kept to -80, -20 or something, those can still create batch effects, which will give you a headache later on. So whatever you choose, whatever kits you purchase, make sure you have enough of those to complete your whole project. Otherwise, you will deal with some batch effects, which are, as anyone here will tell you, are just a pain.
Totally. Thank you. Let me see. There was a follow-up question for Emily. Do you do PCR extraction controls?
Yes, so we use an extraction control at the very beginning. So when we're extracting our DNA, we include a control. It depends on the kit. Sometimes it's PBS, or sometimes the buffer itself acts as the control. So we just wouldn't have a sample included. And then we actually keep the extraction controls and take them all the way through the 16S protocol. And so we sequence our extraction controls and then we'll drop them later on. And the idea would be that they wouldn't have enough reads anyway, so they would kind of automatically be dropped when you're working with your data. And so we do everything the same for our extraction controls. And then the other place that a control does pop up is in the PCR, and those are the non-template controls. And so, you have your triplicates of your PCR reaction, and then you have an extra row that doesn't have your sample, but does have your primer. And so we use that as a way to double check our PCR and make sure that we're not getting extra amplification. So that acts as a control within the PCR, but we don't sequence those NTCs. It's more of a double check when we're doing our gel and then move on as long as it looks good. And then we also use the mock communities. And so those kind of act as our control between sequencing runs. And so those are the different areas where we have a control throughout the protocol.
Thank you. So, Bod, I have a follow-up question for you. So someone was asking about, specifically in this Parkinson's mouse tutorial data set. So they mentioned one of the samples started with something like 16,000 reads, and then is filtered down to 347 features after the denoising step. And just wanted to kind of reiterate, like, what exactly is happening here? Like, are those 347 features reads, amplicon sequence variants? What is going on here?
Yeah, so I would have to actually look at that specifically to give you my impression of what exactly happening. But oftentimes when you are dropping, when you see big drops like that, are either because of the quality of the reads that were in there. And so if a sample had really poor quality sequences for whatever reason, and that could get dropped during the initial filtering phases. If you have a high host untargeted contamination, then they can be dropped during the chimeric removal step. So let me take a look at this particular case, unless that was answered in this particular sample, to get a better idea of what it is exactly that may be happening. Maybe in the meantime, someone else wants to.
Yeah, so while you're looking into that, I'm going to ask Greg really quick. So Greg, a question that we often get at these workshops, and I think it's related to this question here, is with the kind of transition into these modern sequence variant denoising techniques. What kinds of differences do we maybe see in our data sets, say, as opposed to like OTU clustering? Like, are we seeing like similar kinds of signal here? More features, less features? What can we kind of expect?
Yeah, yep, that is a common question. And so, with the new denoising protocols that we're using in like QIIME 2 relative to QIIME 1, we are doing a lot better job of sequence quality control. And so it's really a win-win, because we end up getting higher quality features. So in other words, we're getting less spurious features, or less features that are the result of sequencing error that looks like a real sequence, and we're getting higher resolution. And so if you were to, and I did this a few years ago when we were getting started with developing QIIME 2, I would run the same data set through QIIME 1 and QIIME 2 and compare various metrics. And one thing that I was initially surprised by was that I was getting fewer features with QIIME 2. And I was initially expecting I would see more features with QIIME 2, because we're now looking at the sequence variance, which is a more fine-grained grouping than, say, like a 97% OTU. But it turns out that because we were doing such a better job of sequence quality control, we actually end up getting fewer features with QIIME 2. So that's one difference that you might notice. That's more related to the quality control. All other things being equal, if you apply the same quality control, and you can do this in QIIME 2 if you wanted to, we have some workflows that allow you to do OTU clustering of your sequence variants. In that case, you would end up with fewer, say, 97% OTUs than you would with sequence variants, which you can also think of as like 100% OTU. You should also expect to see, slightly higher resolution. So what that means is that you may sometimes be getting more specific taxonomic assignments. And so maybe if you were able to classify something at a family level with QIIME 1, you might be getting down to a genus level with QIIME 2. And again, that's because it just higher resolution, higher sequence quality in the final results. The issue previously is that like maybe you would group sequences from different genera, say, into the same OTU, and then you may not observe one of those genera. You might get an assignment just for one of them, or you might only get a family level assignment.
Thanks, Greg. Bod, did you have any follow-up on your investigation?
Yeah, so I was just having a look at this one sample that was brought up. Maybe I can share my screen here to see if we can just look at it as a group. Give me 1 second. Okay. Hoping that worked and people can see my screen. Okay. So I think this was the sample that was mentioned. Without, from the top of my head, I don't actually know what the sample is. I'm assuming the HC is for healthy control. And so this is what they were referring to, is that, we started out with 16,000 reads, about 10,000 reads were kept after the filtering. And all of a sudden there's a drastic drop down to 347. Now you can see that this is a very much so an outlier compared to all the other samples where, we not only keep a larger portion of our initial read, but also we denoise most, if not all of them. And so this is, in a situation like this, I would be very hesitant to accept this sample, even if, let's say there was 1,000 reads there. I would definitely take a look at this one particular sample on its own, so you can either import it on its own and look at the quality filter, or if you're more familiar with other sort of quality control tools in R or otherwise, you could look at that one particular sample. And I bet what you'll find is that either the sequencing, the quality scores were really low, or a lot of the sequences had ambiguous nucleotides in, so they were just dropped. That is something Data 2 doesn't like. So an ambiguous nucleotide is, and it basically shows up as a capital letter N, and it signifies that that sequence was not confidently placed as any of the 4 nucleotides that we were expecting, or there might be some other sort of post-processing steps taken that would have inserted those in there. So somewhere along the way, something finicky is happening, and Data 2 doesn't like it, so it's dropping a lot of those samples. They could be chimeric that it was, no, it wouldn't be chimeric, because then we would have seen it dropped here. It could be some other source of contamination or something else that the algorithm just wasn't confident in calling those regions, so dropped it. So in those edge situations, it's always nice to look into the samples specifically. But if you have a ton of samples and you're not really worried about losing one, the easy thing would be just to drop it and move on. Hope that helped a little bit.
Thanks for digging into that a bit, Bod. Yeah, when I first was reading the question, I thought we were talking about the overall number of features in the data set, and not an individual sample. So thanks for clarifying that. Let's see. Any follow-ups to that from the control room here? Nope. Okay. So I have another question here, perhaps for Emily. When we conduct 16S sequencing, we usually add negative control, and it is not a big surprise that we see some things identified in those negative control samples. In this case, what do you suggest? Should we remove those control samples from the downstream analysis, or should we remove all the potential false positive bugs from all the other samples? And I suspect we'll have a little bit of discussion amongst all of the instructors here. So.
Yeah, negative control is used for a couple of different things. So if they're saying a negative control, but it's known, then I wouldn't remove it. However, if they're saying negative control more as an extraction control, then I think that is definitely something we can discuss as a group, because if it is a source of contamination from part of your 16S protocol, then that's a different discussion to have. So I'm going to assume that that's what they mean and let everybody else comment on that one.
Thanks for that. Greg, I see you unmuted.
Yeah. Yeah, a couple things. So like in terms of the samples, if these are like extraction blanks or something like that, definitely remove them. They're not something that you want. Remove the samples. They're not something that you want to include in your analyses. And they'll end up just getting in the way when you're doing things like looking at ordination plots or, say, computing beta group significance statistics. As far as removing the features that you observe in those samples from the other samples, that gets into a little bit of dangerous territory, because just because something is showing up in one of your negative controls doesn't mean that it is not something that is actually present in your actual samples, and so you could get into starting to remove something that is actually a key player in those samples. And so I don't recommend removing things that you observe in your negative controls from all samples. I have seen people do that. It's a common question that we get, but I recommend against taking that approach.
Thanks, Greg. Anyone else have anything they'd like to say on the matter?
I would just like to add that I would also consider how much contamination there is. If you have an extraction blank and it's very little, I think that's unavoidable just as part of a sample going through all the processes in the lab. However, if you have a sample where the extraction blank is almost as high as any of the other samples, that's when I think it needs to be considered a little bit more of whether or not you should remove them. So I think it's also a huge case-by-case basis. Yeah.
Cool. Thank you. Is that Mike over there?
Yeah, I just wanted to add something very, very minor, and only because this has happened to me several times. Sometimes samples can just be mislabeled. I had a couple of times where the sequencing facility sent me back and they mislabeled our positive and negative control. So our negative control, we got back all this data and it matched our mock community. So keep that in mind. Just do the very simple sanity check first to make sure nothing's been mislabeled. So, yeah.
Yeah, that's great advice. Thank you. So let's see. My queue is dry right now. So if you asked a question in the last 2 days and we said, hey, we're gonna save this for a general Q&A and we seem to have missed it, please re-ask it and we'll get it put back into our queue here. While I give folks a minute to kind of percolate and think about things, I'm going to follow up on my interview questions here. So I've been asking the question, how did you get started in microbiome bioinformatics? This could be like computer programming or lab side of things or analysis. And Emily, I'd like to just hear your origin story here. How did you get into this?
Yeah, so I work in Dr. Emily Cope's lab, and we're at the PMI at NAU. And I actually was a TA during my undergrad and just ended up TAing for Emily's class and got along with her really well. Told her I wanted to do, I can't remember what I told her at the time, my master's or my PhD. And she said, great, well, I'm looking for a grad student. And so then I applied and I got in and I just kind of slid my way into it. So this was actually my first research experience for the most part, was in microbiome. So I mostly do lab work. However, due to the nature of a PhD, I also do a lot of my analyses with the help of, Greg's group in general. They're very great. So yeah, I would say that's how I ended up getting into it, just kind of by luck.
That is awesome. Yeah, thanks for sharing. Who else is on the call that I haven't asked that question to? I apologize. My memory is crazy. Mike, come on in.
I am here. I feel like I should change the story every time just to keep it interesting, but no. So I didn't have, a very, exciting foray. It was just I was working on my master's doing phylogenetics. And back then, at the time I did it, we were, using DOS to do things. And there weren't all these cool tools like there are now, like to convert between file formats. And back then, there's a lot of sequence formats, tree formats, and no simple tool to interconvert between them. So finally, after wasting 3 or 4 days just to manually edit a few files in the text editor, I finally, took the dive and I started with Perl, I think, learning how to do, basic commands. And after a while, I saw the benefit and power of being able to, instead of taking 5 hours to format a file, it literally would just take a fraction of a second if you wrote the code. And every time I ran that code, I got that 8 hours back, right? So you get all that time back, and it kind of became a little addictive. And you go down a rabbit hole of just wanting to do more code. So that's kind of how I kind of got into it. And then when I was looking for a PhD, I came to Colorado, which is where I met a lot of folks in the microbiome field. And I just kind of merged it, and I had a hybrid project, sounds like kind of like Emily, where I was mostly lab work. And as later into my PhD I went, the more informatics I kind of became. So yeah, that's it.
It's a pretty empowering feeling getting those 8 hours back on those little tasks. It can be addicting. Yeah. Okay, so got a couple of questions that got bumped back up on the list here. Thank you so much to everyone who shared those with us again. One of them here is, let's see, I'm looking at the last one in our queue here. You covered a good example for using denoising and clustering algorithms. I work with soil microbes, often deal with dominant and rare species affecting diversity estimates. Could you please answer this from the dominant and rare species perspective? Which approach works better for a sample with more dominant species and rare species situations? So I think this question came up during the denoising lecture from Bod this morning. Let's see if any of our instructors want to take a stab at this question here.
I guess I can try a stab if that's okay. Well, I won't talk about the denoising part per se, but there's a great paper, maybe somebody could link it if I don't get to it, by Dr. Kathy Luzapone called Qualitative and Quantitative Insights into Microbial Ecology. And in that paper, she highlights the benefits of using basically what amounts to presence-absence like approaches and abundance-based approaches. And if you really care about those rare taxa, you're probably going to lean towards presence-absence based approaches, which I think a lot of this will be talked about later this week. So basically, it's a matter of the algorithm that you're using, whether or not it upweights or downweights based on abundance or presence and absence. And they can tell you different things. So, but if you're looking for just a one-stop shop on how to handle this, they're really kind of, well, I don't think there really is one, but you can stratify your data set and break it out and reanalyze it for various questions of your data. So if you wanted to look at your abundant component of your data, you can analyze it that way. And then if you're looking at just rare taxa, you can pull those out or just look at those. There's a variety of ways to do this, but leave it there because this is kind of one of those rabbit hole questions, I think, but I'm sure others will chime in.
Well, I'll let folks think about that for a couple minutes. I'll move on to another question, and if we have any more thoughts percolate, we can circle back to it. There is a follow-up question related to Bod's Data 2 denoising stats investigation, where he pulled up the denoising stats and kind of talked about that one sample where we lost like almost 16,000 reads in it. And so there was a question about how can we investigate the quality of that individual sample. If you don't mind, I'll take a stab at answering this really quick. So control room, I'm going to share a screen. Which screen is, that is the question. Let's see, is that shared? Yes, okay. So I have the QIIME2 docs pulled up. You'll notice this is a common theme today. I have kept a tab for the QIIME2 docs up basically all day. And over here on the sidebar, there's a table of contents, and what I'm clicking on is plugins and then available plugins. And this has the help text for every one of the commands in the kind of core distribution of QIIME2. And for this particular question about how could I look at the quality scores for one particular sample, I think the easiest way that you could do that right now in QIIME2 without jumping out of QIIME2 would be to filter the samples first. So you could run this command. We won't be covering this command specifically in this workshop here, but you could run this command to filter out so that you only had the sample or samples in question, and then rerun that demuck summarize question or command that came up before. And so then you would just be seeing the quality score profile for that particular sample or group of samples if you chose to filter more than one. Otherwise, if you wanted to look at this outside of QIIME2, I've used FastQC for this a couple of times. That's a pretty easy tool to download and install, and I think it's a little Java app, and that's another good way to quickly look at that. Any follow-ups from the crowd here? No. Okay. Let's see, we have a couple of minutes left here in today's session. Hopefully we can get through the rest of these questions. I have a question here about mock communities. Do you include a mock community control on each plate? I suspect everyone here has some thoughts on this. Emily, do you want to go first, or? Okay, yeah.
So I'll share our lab experience, and then we can have comments on other thoughts. We have 4 mock communities that we use on all of our sequencing runs. The way that I like to set it up is I put 1 mock community per extraction. That's just my preference. And then they all get sequenced and they all end up getting dropped. But we have 3 mock communities that I guess would be considered positive, and then a control mock community that would be more of our negative. And that's what we use.
Can I? So I also have a kind of a question, but I'll comment first, and then I'll ask of the instructors and the group. Sometimes people get worried when their mock community doesn't come back as it's supposed to, right? You order a mock community for a company, and with the equal abundance, I guess, of each of those groups. And we often sometimes see questions that, well, do I have to redo my analysis because I ran 2 mock communities on my run, but they don't match what I have? So keep in mind your mock communities will also be subject to primer bias, right? So a primer might preferentially amplify one of those mock community members better than the other, and that's okay. I know our group, we tend to look for consistency of what is returned community across the plates. So as long as we're seeing this consistently, okay, yep, we're getting more of this and a little of this each time, and it's consistent, that's one way you can think about how to use the mock community. I just wanted to bring that up as an insight, but I'm sure others have other ways they leverage that.
One comment, if you have mock communities and you have access to them, definitely sequence them, keep them. There's a whole other dimension of microbiome data that relates to compositionality, and this is something I think maybe Evan might discuss in one of his presentations or talks, or at least I know he's definitely in the conversations of that. So this is all based on a paper by, again, Ben Callahan, Amy Willis, and Mike McLaren that I'll provide a link to, and I definitely recommend everyone having a read for it. So it's one of these situations where we probably should be using our mock communities to, in some ways, fine-tune or calibrate our sequencing, but I don't have a good solution or exactly how to do this. So I'll just provide the link so at least you sort of are in the know about the problem that arises with a lot of microbiome analyses, and it's really nicely highlighted in that paper. But at the end of the day, until we figure out what is the best way to use our mock communities, having them, even if you don't end up using them directly, having them is going to be a massive boost when down the line we have sort of the bioinformatic pipelines and the knowledge of how to use them properly. And of course, for all the other benefits that everyone just mentioned as well.
Thanks for that. Greg, do you have anything to add to the topic?
Nope, I don't think so. Not right now. Most of my sequencing runs these days were including mock communities in the run. And, like Mike was saying, I'm really often using those to look for consistency across runs or across samples.
All right. Well, thank you, everyone. There is one more question in the queue here that's about some specifics to trimming with CutAdapt. I think perhaps we will follow up directly in line with you, because this does seem like maybe it's a little bit of a specific question, and then if we still if we have some time tomorrow, we can circle back to it. So I think with that, I'm going to wrap us up here for the day. Thank you, everyone, for your fantastic questions. Thank you to all of the instructors for making time to be with us today. It was a pleasure. So just to briefly recap what we did today. So Emily first walked us through some 16S lab protocols. Then Bod and Chloe walked us through importing and demultiplexing sequences, both the theory and the practice. And then Bod and Chloe again walked us through denoising using our Parkinson's mouse tutorial data set. And then finally, Mike and Chloe covered the phylogenetic reconstruction side of things. So if you've been following along with the tutorial, you should be to a point where you have a phylogenetic tree created, as well as a feature table and some representative sequences. Tomorrow, we are going to kick the day off with some discussion about taxonomic characterization of our samples or the features. And we will be spending actually quite a lot of time tomorrow on the interactive tutorial. So looking forward to that. I will post the day 3 videos here in just a few minutes. So if you would like to work ahead or pre-watch any of these lectures or tutorial segments, that would be a great time to do that. Otherwise, we will see you back at the same time tomorrow. So that is 9 a.m. Mountain, and we will be ready to go then. Thank you.